Welcome to IMAT Tutor. In this high-yield overview, we are launching Lecture 10 of our IMAT Biology series, where we dissect the entire human circulatory system. Think of the circulatory system as the ultimate biological infrastructure, a sophisticated network of blood, vessels, and the heart, working in unison as the fundamental machinery that sustains every single cell in the human body. To master this high-yield IMAT topic, we have broken down our roadmap into 8 essential subtopics that you must know inside and out. Our journey begins with heart anatomy and structure, where we will examine the muscular walls, chambers, and valves that dictate unidirectional blood flow. From there, we transition into the cardiac cycle, mapping out the precise mechanical phases of systole and diastole. Of course, the heart cannot pump without an electrical trigger, which brings us to the conductive system, the specialized myogenic tissue coordinating the rhythmic heartbeat. Once the blood leaves the heart, it navigates the complex pathways of our circulation circuits, specifically separating the oxygenating pulmonary circuit from the nutrient-delivering systemic circuit. To understand how this fluid moves, we will analyze the structural histology of the blood vessels, contrasting high-pressure arteries with high-capacitance veins and exchange-heavy capillaries. This structural layout leads directly into the dynamics of blood pressure regulation, where we will break down the neural, hormonal, and renal mechanisms that maintain homeostatic perfusion pressure. Next, we dive deep into the fluid itself by evaluating the blood components, distinguishing the cellular elements from the lipid plasma. Finally, we wrap up the lecture by investigating homeostasis and lymph, covering the vital cascades that prevent blood loss alongside the lymphatic network that maintains tissue fluid balance. Let's dive in. It's gonna be a long, complete lecture. After watching this, you don't need to look for anything else anywhere for the topic of the circulatory system. To truly grasp how cardiovascular dynamics operate from the first principle, imagine a completely blank whiteboard. Let's start building the human circulatory plan from the ground up. Why do we need a specialized transport system in the first place? It all comes down to the physical limits of fusion. Let's say this is a single-celled organism, or let's imagine these are some clusters of cells. For a single-celled organism or a tiny cluster of cells, simple passive diffusion across a cell membrane is more than enough to exchange gases and nutrients. But as organisms evolve into large, complex, multicellular bodies, the distance between the external environment and the innermost cells grow vastly larger than the microscopic range over which passive diffusion is efficient. We require bulk flow, a hydrodynamic connective system powered by pressure gradients to bridge that distance. Let's draw this bulk flow mechanism as a complete circuit. First, we establish a specialized fluid medium to carry these molecules, that is, blood. To contain this blood and direct its pathway to prevent it from simply spilling out into the interstitial spaces, we draw a continuous, unbroken network of muscular and endothelial tubes. That means the blood vessels. Because fluid naturally meets resistance as it rubs against these vessels, it will eventually stop moving due to friction. That means the further it tries to go, it will stop due to the friction that is caused by the walls of the blood vessels. To keep it circulating, we must introduce a mechanical pressure source. For that, let us draw a central muscular pump right in the middle of our diagram. That means the heart. Because the fluid never leaves this continuous loop of heart and vessels, we call this a closed circulatory system. Now let's look at how this fluid loops. If we were to draw a simple single circuit loop where blood goes from the pump, that means the heart, to the gas exchange organ, let's say these are the lungs, then let's say straight to the body tissues, and then back to the pump. The fluid pressure would drop drastically across the microscopic exchange vessels of the respiratory membrane. By the time that blood reached the rest of the body, that means from here, its pressure would be far too low to ensure effective perfusion. Perfusion means the exchange of gases. To solve this physiological design flow, humans utilize a double circulatory system, splitting the layout into two distinct loops arranged in series. Let's sketch these two circuits side by side to understand the exact mechanics. We represent the heart not as a single uniform block, but as four distinct muscular chambers, acting as two separate pumps operating simultaneously. On the right side of the diagram, we draw the right pump. The upper one is right atrium and the lower one is right ventricle. The right atrium receives systemic deoxygenated blood from tissues and ejects it under relatively low pressure into the first circuit, and the circuit is pulmonary circuit, and right ventricle does the job of ejecting this blood to the lungs. In the lungs, carbon dioxide is dropped off and fresh oxygen is picked up. We saw this whole process in our respiratory system lecture. The freshly oxygenated blood then returns to the heart, but the catch is it doesn't return to the right side. Instead, it returns to the left side of the heart. To be more precise, it returns to the left atrium of the heart. From left atrium, the blood first goes to the left ventricle. This left ventricle with the oxygenated blood generates a massive hydrostatic force to drive it into the second loop. And the second loop is the systemic circuit. Left ventricle propels blood out to the rest of the body to sustain metabolically active tissues like this. Body means the organs, the skin, every part except lungs. As these tissues consume oxygen and produce metabolic waste, the blood becomes deoxygenated again and flows right back into the right atrium, as we were saying before, and it completes the master loop. Because these two circuits are strictly arranged in series, every single drop of blood must sequentially pass through both loops. The volume of blood pumped by the right ventricle per minute must exactly equal the volume of blood pumped out by the left ventricle per minute. That means the cardiac outputs must be identical. Now that we have visualized this entire physiological mechanism evolving dynamically on our whiteboard, let us transition directly to our text and anchor these illustrated concepts to the exact keywords and definitions you will encounter on your exam. Starting off with the definition, humans possess a closed double circulatory system consisting of three interconnected components. First, the heart, which acts as a dual muscular pump, the blood vessels, which form the continuous conduits, and the blood, which serves as the fluid transport medium. The system is divided into two distinct anatomical circuits, arranged sequentially in series, the pulmonary circuit and the systemic circuit. The right side of the heart contains chambers dedicated to receiving deoxygenated blood from the systemic tissues and pumping it directly toward the lungs for gas exchange. The left side of the heart consists of chambers that receive freshly oxygenated blood coming from the lungs and pump it with high pressure out to the rest of the body. Because the pulmonary and systemic loops are positioned in series, the volume of blood ejected per unit time, that means the cardiac output, must be absolutely equal from both right and left ventricles to prevent fluid from backing up. Pay critical attention to these high-yield exam traps. Examiners love to test your understanding of what in series actually means. They will try to trick you into thinking that blood can choose to go to either the systemic circuit or the pulmonary circuit, as if they were arranged in parallel like roads branching off a highway. Remember, in series means all blood must pass through both circuits sequentially, not one or the other. Another classic trap is misdefining blood vessels based purely on the oxygenation state of the blood they carry. Students often fall into the trap of stating that all arteries carry oxygenated blood and all veins carry deoxygenated blood. This is fundamentally false. The absolute unchanging anatomical definition is that arteries always carry blood away from the heart, while veins always carry blood toward the heart, regardless of the gas saturation level. That's why it is written that arteries carry blood away from the heart and not always necessarily oxygenated blood. If we want to have some take-home message from this slide, then we must remember the pulmonary artery is the only artery in the postnatal human body that carries completely deoxygenated blood, moving it away from the right ventricle into the respiratory exchange tissues of the lungs. Conversely, pulmonary vein is the only vein in the human body that carries highly oxygenated blood, transporting it directly from the lungs back toward the left atrium of the heart, as we have seen in the diagram previously. If you want IMAT biology notes that actually help you score, that are not bulky textbooks, not random summaries, but exam-focused, trap-oriented, high-yield PDFs, the link is in the description. Study smart, not hard. To truly grasp heart structure from first principles, let's build the macro and micro architecture of this master pump from scratch. If we want a pump capable of generating a powerful force, we first need to build its structural container. And that brings us into pericardium, that is the covering of the heart. To understand it better, we have zoomed into pericardium and we have shown different layers over here. The outermost layer of pericardium is tough, unyielding connective tissue barrier called the fibrous pericardium, which prevents overfilling of the heart chambers. Just deep to fibrous pericardium, we will have serous pericardium, one layer over here, another here. So evidently, serous pericardium has two distinct layers of its own. The outer one, we mean this, lines the underside of the fibrous wall as the parietal layer of serous pericardium. Next, we look at an explicit fluid space, the pericardial cavity, which is filled with a thin film of lubricating serous fluid. This fluid is an absolute mechanical necessity, minimizing frictional drag as the heart violently contracts and expands. Immediately deep to this cavity, the serous pericardium forms our outermost cardiac wall layer, the epicardium. Moving inward, we will see the thickest, most energy-demanding layer of the heart wall, that is this region. This is called the myocardium. This layer is composed of highly specialized cardiac muscle tissue arranged in spiral bundles. Why do we need this unique muscular configuration? Because when these cells depolarize and shorten, they wring the heart like a wet towel, maximizing the volume of blood ejected during contraction. Finally, to line the inner hollow interior of this muscular pump, there is a glistening, ultra-smooth layer of simple squamous epithelium resting on connective tissue, highlighted here, and it is called the endocardium. This innermost layer forms an unbroken sheet that lines all internal chambers and completely coats the heart valves, ensuring that flowing blood never encounters a rough surface. Now let's partition this hollow muscular shell into an efficient four-chambered layout. We draw a vertical muscular wall, this is called the septum, right down the middle, which separates the heart into right and left side. Then we divide the upper and lower portions with another horizontal line. The upper chambers are the receiving rooms, left atrium and right atrium. The lower chambers are the powerful discharge pumps, the right ventricle and left ventricle. When we would look at the ventricles of the heart, we will see structural divergence between these two ventricular walls. Left ventricular wall, it will be significantly thicker than the right ventricular wall. And what is the reason behind it? It all comes down to the physical workload imposed by fluid resistance. The right ventricle only needs to push blood into the short, low resistance pulmonary circuit to the lungs, operating under a low pressure gradient. The left ventricle, however, must generate an immense hydrostatic force to overcome the massive systemic vascular resistance of the entire body. Therefore, the left ventricular myocardium must be vastly thicker to sustain this highest systemic pressure. Now we will be looking at the cross-section of the heart to trace the dynamic sequence of blood flow through these chambers. On the right side, three massive systemic channels empty their deoxygenated contents into the right atrium at first. From the upper side of the body, we have superior vena cava, and inferior vena cava drains the lower body. The third one is coronary sinus. To trace the coronary sinus, we need to look at the posterior side of the heart. That means the back of the heart. After rotating the image, we see the third one as coronary sinus. This coronary sinus collects spent blood from the heart's own coronary circulation. From the right atrium, blood passes into right ventricle. Upon contraction, the right ventricle ejects blood into the pulmonary trunk. This structure is the pulmonary trunk we're talking about. To have a clear view of the pulmonary trunk, we have changed the diagram. This pulmonary trunk bifurcates and gives rise to left pulmonary artery, and another one would be right pulmonary artery, which we cannot see. But let's imagine this is the right pulmonary artery arising from pulmonary trunk. These two pulmonary arteries carry blood to the lungs. The left bringing blood to the left lung and the right artery bringing blood to the right lung. After completing gas exchange within the pulmonary capillaries, the freshly oxygenated blood must return to the heart. To clearly understand this process, we will look at this specific diagram. Pulmonary veins will perform the task of bringing back blood to the heart from the lungs. Two left pulmonary veins, 1 and 2, and two right pulmonary veins, all will converge to empty their oxygenated contents into the left atrium. As you can see, they are converging here in left atrium. From left atrium, blood flows into left ventricle. With the final high pressure contraction, the left ventricle drives this blood through the massive arched aorta. This is the aorta that we're talking about. This aorta will distribute oxygenated fuel to the systemic tissues of the entire body, where it will eventually return to right atrium to begin the sequence anew. Now that we have visualized this entire structural blueprint and blood flow pathway evolving dynamically from first principles, let us transition directly to a text and anchor these illustrated mechanisms to the exact keywords, sequences, and clinical pitfalls you will encounter on your exam. To formalize the framework we just illustrated, the human heart is classified as a four-chambered muscular organ, structurally divided into a right atrium, right ventricle, left atrium, and left ventricle. Correlating with our explained diagrams, the cardiac wall consists of three histologically unique layers: the outermost epicardium, the intermediate muscular myocardium, and the innermost endothelial endocardium. The outermost superficial layer, that is epicardium, is anatomically identical to the visceral layer of serous pericardium that we place just deep to the fluid-filled pericardial cavity. Myocardium forms the highly contractile intermediate layer consisting entirely of cardiac muscle cells, functioning as the primary engine responsible for generating the heart's mechanical pump force. Endocardium is the innermost layer composed of a smooth endothelium lining all internal chambers and valves to guarantee smooth fluid flow. Reflecting the pressure workloads we established, the muscular wall of the left ventricle is significantly thickest because it must pump blood against high systemic vascular resistance, whereas the right ventricle wall is much thinner due to the lower pressure demands of the pulmonary circuit. In the mechanism section, our flow sequence begins as the right atrium acts as the central collecting chamber for deoxygenated blood, receiving systemic input directly from three vessels: the superior vena cava, the inferior vena cava, and the coronary sinus. From there, blood travels sequentially from the right atrium to the right ventricle, which then contracts to drive fluid out through the pulmonary trunk and into the pulmonary arteries to reach the lung capillaries. Once gas exchange occurs, freshly oxygenated blood exits the respiratory tissues and travels through a total of 4 independent pulmonary veins to enter the left atrium. To complete the master loop, blood moves from the left atrium to the left ventricle, which powerfully ejects the fluid directly into the aorta for distribution across the entire systemic body network. Pay critical attention to these high-yield exam traps to protect your score, as examiners frequently exploit the complex arrangement of these venous inputs and vessel counts. They love to test your detailed knowledge of the heart's intrinsic venous drainage by asking exactly where the coronary sinus deposits its deoxygenated blood. Because it carries highly spent blood from the heavy left ventricular muscle mass, students frequently fall into the trap of guessing that it drains into the left atrium. Do not fall for this. The coronary sinus strictly drains its deoxygenated blood back into the right atrium alongside superior vena cava and inferior vena cava, not into left atrium. Another frequent trap involves the precise count of the pulmonary vessels returning to the left pump. Because students are highly conditioned to think of bilateral body structures occurring in basic pairs, examiners will write sneaky multiple choice questions stating that blood returns from the lungs via 2 pulmonary veins, one for each lung. Pay strict attention here. There are exactly 4 pulmonary veins, 2 originating from the left and 2 originating from the right lung, and all open independently into the left atrium. To understand how the heart enforces unidirectional flow, we will build the concept of mechanical architecture of cardiac valves completely from scratch. When a muscular chamber like ventricle contracts, it generates a massive hydrostatic pressure gradient. Fluid naturally wants to move from an area of high pressure to an area of low pressure. Without structural check valves, this pressure would drive blood backward into the low pressure atria, just as easily as it drives it forward into high pressure arteries. To prevent this fatal backflow, the heart utilizes 4 specialized fibrous valves operating purely on passive pressure differentials. To get a better understanding of the valves, we will look at this diagram where the atria has been removed to see the valves clearly. Valve number 1, 2, 3, and 4. In total, 4 valves are seen in this diagram. Now let's split these 4 valves into 2 functional pairs based on their anatomical positioning. First, the atrioventricular or AV valves. These valves fit directly between the atria and ventricle. On the right side of the diagram, we see tricuspid valve. This is a tricuspid valve. This is a valve composed of 3 distinct fibrous flaps or cusps, 1, 2, and 3. Mirroring this on the left side, separating the left atrium from the left ventricle, we see bicuspid valve with exactly 2 cusps. The bicuspid valve, universally referred to as the mitral valve, has been named mitral due to its resemblance to a bishop's mitre. Let's look at the mechanical problems these AV valves face. During ventricular contraction or systole, the pressure inside the ventricle skyrockets. This immense upward force pushes the cusps together to seal the doorway. Let's look at the mechanical problem these AV valves face. During ventricular contraction or systole, the pressure inside the ventricle skyrockets. This immense upward force pushes the cusps together to seal the doorway, but it is so strong that it threatens to blow the valves inside out, collapsing into atria. To solve this physiological engineering challenge, tendon-like cords called chordae tendineae are attached to the free edges of the valve cusps. In this diagram, you see the cut section of heart, and the chordae tendineae are attached to the free edges of the valve cusps over here. Then anchoring these cords to the ventricular floor, we have specialized muscular pillars projecting from the myocardium, the papillary muscles. These are the papillary muscles, and they are attached to the wall of the myocardium, as you can see here. It is vital to realize the exact sequence here. When the ventricles contract, the papillary muscles contract simultaneously, pulling the chordae tendineae taut. These cords do not close the valves. Rather, they act as active tension tethers that prevent the passive valve cusps from swinging backward into the atria under peak pressure. Imagine these are the cusps of the valve, they will only open in one direction. Now let's look at the second pair of check valves, those guarding the exit points where blood leaves the ventricles to enter the great arteries. These are the semilunar valves, one over here, another here. Guarding the exit from the right ventricle into the pulmonary trunk, we will find the pulmonary semilunar valve. Guarding the exit from the left ventricle into the ascending aorta, we have the aortic semilunar valve. Structurally, these valves have exactly three pocket-like crescent moon-shaped cusps. Unlike the AV valves, these semilunar exits do not experience a violent structural threat of inversion into a wider chamber, so it doesn't draw any chordae tendineae or papillary muscles here. Instead, when the ventricles relax during diastole, the arterial blood pressure becomes higher than the ventricular pressure. This back-shifting blood physically catches in the pocket-like semilunar cusps, inflating them like tiny parachutes until their edges meet in the center, snapping the vessel shut and preventing blood from slipping back into the relaxing ventricles. Now that we have visualized this entire mechanical check valve system evolving dynamically from first principles on our diagram segment, let us transition directly to our text and anchor these illustrated mechanisms to the exact keywords, sequences, and clinical pitfalls you will encounter on your exam. To formalize the framework we just drew, the heart contains four distinct valves, divided into two functional pairs, two atrioventricular valves and two semilunar valves, all ensuring unidirectional blood flow. Looking at the key features table, we first see tricuspid valve. This specific AV valve is located on the right side of the heart between right atrium and right ventricle and is characterized by having exactly three fibrous cusps. Mitral or bicuspid valve is located on the left side of the heart between left atrium and left ventricle and is uniquely characterized by possessing only two structural cusps or flaps. Then if we talk about the pulmonary and aortic semilunar valves together, first, we need to say the pulmonary valve guards the exit of the right ventricle into the pulmonary trunk, while the aortic valve guards the exit of the left ventricle into the aorta. Both consist of three crescent-shaped pocket-like cusps. Correlating with our diagram sequence, the free edges of the AV valve cusps are physically anchored by fibrous cords called chordae tendineae, which are routed directly into the ventricular walls via specialized papillary muscles. During ventricular systole, the papillary muscles contract alongside the ventricular myocardium, pulling the chordae tendineae taut to prevent the valve flaps from prolapsing or inverting backward into the low-pressure atria. During ventricular diastole, as ventricular pressure drops below arterial pressure, backflowing blood collects within the pocket-like cusps of the semilunar valves, forcing them closed without requiring any supporting cords or muscular anchors. Pay critical attention to these high-yield exam traps to protect your score on the IMAT. Examiners love to exploit the most common anatomy error by swapping the sides of the AV valves. Always remember that tricuspid equals right and has three cusps, while the mitral equals left and has only two cusps. Do not mix up their locations. Another major trap focuses on the number of cusps in the exit tracts. You must remember that all semilunar valves have three cusps, not two. Students frequently misidentify them as bicuspid because they confuse them with mitral valve. Looking at the examples, we first talk about systolic murmur. A systolic murmur occurs during a ventricular contraction. This happens either because an AV valve fails to seal tightly when it should be completely closed, which leads to mitral regurgitation, or because a semilunar valve fails to open fully when blood is trying to escape, resulting in aortic stenosis. And a diastolic murmur occurs during ventricular relaxation and filling. This clinical sound is generated either because an AV valve is narrowed and cannot open smoothly to let blood in, resulting in mitral stenosis, or because a semilunar valve fails to remain closed, causing blood to leak back into the heart, presenting as aortic regurgitation. Our next topic deals with cardiac cycle. To master this precise timeline of a single heartbeat, let us establish a blank conceptual map of the cardiac cycle based entirely on the first principles. A cardiac cycle is a continuous mechanical loop governed by a simple rule of physics. Fluid moves down a pressure gradient, and that movement is strictly channeled by the opening and closing of our cardiac valves. To quantify this system, we introduce time. One full cycle lasts approximately 0.8 seconds if we consider the standard resting heart rate to be 72 beats per minute. Let us break this time block down into two primary mechanical states. First one is diastole, where the ventricles are relaxing and filling, and systole, where the ventricles are contracting and ejecting fluid. Let us position our first baseline phase on the conceptual map, ventricular filling, which occurs during late ventricular diastole. At this moment, the ventricular muscle is completely relaxed, and the pressure within the ventricular chamber is lower than the pressure within the atria. That means the pressure inside atria is greater than the pressure inside the ventricle. We proportionally draw the letter P to indicate the difference in pressure. This pressure gradient forces the atrioventricular valves to snap open, indicated by this arrow. Passive blood rushes from the atria into the ventricles, causing the ventricular volume to scale upward to its maximum limit. Toward the end of this phase, the atria undergo active contraction, that is atrial systole, to squeeze the final remaining volume into the lower chambers. The dark shade represents the blood, and the blood is coming toward ventricle, and the arrival of blood finalizes in atrial contraction phase. The total amount of blood resting in the ventricle at the absolute end of this filling phase is defined as the end diastolic volume, or EDV. Now let us introduce the transition into the ventricular systole. The ventricular myocardium begins to contract forcefully, indicated by these arrows. As a direct result, pressure inside the ventricles spikes sharply upward. The instant this internal pressure exceeds the atrial pressure, blood moves backward against the atrioventricular valves, indicated by the black arrows over here, and it snaps the valves completely shut. Crucially, at this exact millisecond, the ventricular pressure has not yet risen high enough to overcome the massive arterial pressure resting inside the aorta and pulmonary trunk, meaning the semilunar valves remain firmly shut. The semilunar valves are here, one in this place, the other one in this area. Because all four valves are completely closed, blood cannot enter or leave the chamber. This means that as the myocardium contracts, the internal pressure rises vertically, but the fluid volume remains completely unchanged. We define this highly dynamic state as isovolumetric contraction. Let us follow the pressure curve as it continues climb. The moment intraventricular pressure breaches the arterial threshold, it forces the semilunar valves to open. This valve and this valve, both of them are semilunar valves. This initiates the ventricular ejection phase, seen in the next diagram. Blood is driven out of the heart and into the great vessel. One of the great vessels is pulmonary trunk, another one is aorta. This is the pulmonary trunk giving off two branches, and this one is the aorta, which is arching moving forward. When the blood goes out of the heart and into the great vessels, the ventricular volume drops rapidly. However, the heart never empties itself completely. The residual blood left behind in the ventricle at the absolute end of this ejection phase is defined as the end diastolic volume, or ESV. The actual volume of blood successfully propelled out of the heart per beat is the difference between what we started with and what we left behind, a crucial metric known as the stroke volume, or SV. So the stroke volume SV would be end diastolic volume. That means the blood remaining after diastole minus end systolic volume that we have just discussed. And to evaluate the mechanical efficiency of this pump, we calculate the percentage of total blood ejected relative to the starting volume, a formula known as the ejection fraction. Ejection fraction EF is equal to stroke volume divided by end diastolic volume times 100. And if you multiply the stroke volume with heart rate, we calculate the total volume circulated every single minute. And that is the cardiac output we write as CO. Under the Frank-Starling mechanism, the heart naturally matches this output to venous return. That means an increased return stretches the cardiac muscle, generating a more forceful contraction and an increased stroke volume to maintain circulatory balance. Finally, systole ends and the ventricles begin to relax, dropping internal pressure precipitously. The instant ventricular pressure falls below arterial pressure. That means the pressure inside the ventricles is decreasing gradually. As the pressure decreases, blood tends to backflow, and this backflow catches the semilunar valves, one over here, another one here. And these valves are snapped shut. That means they are closed due to the pressure gradient. Because ventricular pressure is still higher than the atrial pressure, that means the pressure inside the ventricle is still higher than the pressure inside the atria. That's why we light. Finally, systole ends and the ventricles begin to relax, dropping internal pressure precipitously. The instant ventricular pressure falls below arterial pressure. That means the pressure inside the ventricles is decreasing gradually. As the pressure decreases, blood tends to backflow, and this backflow catches the semilunar valves, one over here, another one here. And these valves are snapped shut. That means they are closed due to the pressure gradient. Because ventricular pressure is still higher than the atrial pressure, that means the pressure inside the ventricle is still higher than the pressure inside the atria. And that's why we're writing tiny p's inside atria. Due to this pressure difference, the AV valves, one over here, another one here, they remain closed. Once again, all four valves are sealed, causing internal pressure to crash while volume remains perfectly constant. This is isovolumetric relaxation, which terminates when ventricular pressure drops low enough to reopen the AV valves, restarting our filling sequence. Now that we have mapped out this entire mechanical timeline from first principles, let us transition directly to our text and anchor these concepts to the exact sequence, formulas, and high-yield traps on the anchor slide. To formalize our timeline, one complete heartbeat is defined as a continuous sequence consisting of four distinct mechanical phases: filling, isovolumetric contraction, ejection, and isovolumetric relaxation, lasting approximately 0.8 seconds at a resting heart rate of 72 beats per minute. Looking at the mechanism, the first phase is ventricular filling, that means diastole. People might get curious that why are we talking about ventricles? Why atria is not considered in this sequence? And that is because ventricles do the heavy lifting of pumping the blood to the whole body. Isovolumetric contraction marks the first phase of ventricular systole. In this phase, all four valves are closed. As the muscle contracts, intraventricular pressure rises sharply while volume remains completely constant. Then ventricular ejection is triggered when pressure overcomes arterial resistance. When the pressure is overcome, the semilunar valves open, while AV valves are closed, causing ventricular volume to drop to its minimum point, which is the end systolic volume, which averages 50 to 60 milliliter at rest. Let me give you a few more nuggets of knowledge before we talk about isovolumetric relaxation. The first one is the stroke volume, or SV, and the cardiac output together. Stroke volume represents the actual amount of fluid ejected per beat, which is 70 milliliter at rest, calculated as end diastolic volume minus end systolic volume. Multiplying this by the heart rate yields the cardiac output, averaging approximately 5 liters per minute at rest, which can climb to 20 to 25 liters per minute in a trained athlete during exercise. Then we talk about ejection fraction, and this metric represents the percentage of blood pumped out, and we calculate it by stroke volume over end diastolic volume times 100. With a normal range of 55 to 70%. A value below 40% indicates clinical heart failure. Now coming back to isovolumetric relaxation. This occurs during early ventricular diastole, and all valves are closed once more as the muscle relaxes, causing intraventricular pressure to fall while volume remains completely constant. Now pay critical attention to these high-yield exam traps to protect your score, as examiners love to test your absolute clarity on the valve status during the cycle. A classic trap is suggesting that blood is moving or that certain valves are open during the isovolumetric phases. Remember the strict physical rule. During both isovolumetric phases, which involve isovolumetric contraction and isovolumetric relaxation, all four valves are simultaneously closed, making volume change impossible. Another major trap involves the precise mechanical origin of our heart sounds. S1 and S2. Students often mistakenly believe that heart sounds are caused by the active opening of valves. Memorize this rule. Heart sounds are exclusively caused by the sudden passive closure of valves. S1 is the sound of the AV valves closing at the start of systole, and S2 is the sound of the semilunar valves closing at the absolute end of systole. To master clinical acoustic relationship on your exam, the brief time interval between hearing sound S1 and sound S2 corresponds directly to ventricular systole. Conversely, the longer interval between S2 and S1 represents ventricular diastole. This S1 is the subsequent S1 that is generated after this S2. Another pearl I want to share is the tachycardia trap. While moderate exercise safely elevates cardiac output, extremely high pathological heart rate activate the tachycardia trap, where diastolic filling time is shortened so drastically that end diastolic volume plummets, causing both stroke volume and overall cardiac output to paradoxically fall. This segment might look repetitive, but to master the quantitative core of CVS dynamics, CVS stands for cardiovascular system, let us establish a blank conceptual map of cardiac volumes and performance metrics based entirely on first principles. The heart is fundamentally a hydrodynamic displacement pump whose baseline output is determined by two shifting physical boundaries: the maximum volume it can hold and the residual volume it cannot get rid of. To mathematically define the system, we focus on the lower pumping chambers during the transition from relaxation to contraction. Let us position our first primary volume boundary on the conceptual map, and diastolic volume, or EDV. This value quantifies the total amount of blood that has filled the ventricular chamber by the absolute end of diastole, serving as the fluid starting line before contraction begins. As we have said earlier, end diastolic volume is around 120 to 130 milliliter at rest. Next, we establish the opposite boundary, the end systolic volume, or ESV. When the ventricular myocardium contracts during systole, it generates immense force. Yet, it is physically impossible to obliterate the entire chamber space. A specific residual volume remains trapped inside at the absolute end of ejection, shown by this blood volume over here. By analyzing these two boundaries, we derive the actual mechanical displacement of the pump. The difference between the volume we started with and the residual volume left behind dictates the exact amount of blood successfully propelled out into the circulation per single stroke. We define this fundamental performance metric as the stroke volume. So stroke volume is end diastolic volume minus end systolic volume. To scale this up and evaluate the continuous performance of the cardiovascular network over time, we introduce a temporal variable, which is heart rate. Heart rate measures the frequency of contractions per minute by taking the volume ejected per single beat. That means by taking stroke volume and multiplying it by heart rate, we establish the total volume of blood moving through a circuit every 60 seconds. And this master volumetric rate is defined as the cardiac output. Now let us introduce a metric to measure the relative efficiency of this ejection phase. Not all ventricles are built the same, and looking at stroke volume alone does not tell us what portion of the available blood was successfully utilized. To solve this, we establish a proportional ratio known as the ejection fraction. This formula divides the stroke volume by total starting and diastolic volume, multiplying by 100 to express the pump's performance as a clean percentage. Let us look at how this fluid displacement responds to changing physical demands. The heart possesses an intrinsic mechanical law to match its output to varying workloads, known as the Frank-Starling mechanism or the Frank-Starling law. When venous return increases, a larger volume of blood flows into the heart, stretching the myocardial fibers. Within homeostatic limits, the increased stretch optimizes the alignment of the contractile unit of the cardiac muscles, which produces a significantly more forceful contraction that naturally scales up the stroke volume. However, if heart rate escalates pathologically, a physical timing conflict emerges. Because diastole is a time-dependent filling process, an excessively high heart rate compresses the available relaxation window, severely shortening filling time. Consequently, the pump cannot accumulate adequate fluid, causing the starting volume to fall, which ultimately drags down both stroke volume and total cardiac output. Now that we have mapped out these volumetric variables and equations, let us transition directly to our text and anchor these concepts. Formalizing the definitions on slide 5. And diastolic volume refers to the total volume of blood in the ventricle at the end of filling, averaging approximately 130 milliliter at rest. And systolic volume, ESV, represents the residual volume remaining in the ventricle after contraction is complete, sitting at approximately 50 to 60 milliliter at rest. Stroke volume, or SV, is calculated explicitly as the difference between our two primary boundaries. That's why it is written as N diastolic volume minus N systemic volume, and this represents the volume of blood ejected per single heartbeat and averages approximately 70 milliliter. Cardiac output represents the total blood volume circulated every single minute, calculated as stroke volume times heart rate and averaging approximately 5 liter per minute at rest. Ejection fraction is a parameter that quantifies pumping efficiency of the heart as a percentage, and the formula is stroke volume over N diastolic volume times 100, with the standard healthy normal range established between 55 to 70%. When ejection fraction drops below 55%, it serves as a critical clinical marker for impaired ventricular function and heart failure. And Frank-Starling law is an intrinsic mechanical principle that states that an increased end diastolic volume leads directly to an increased stroke volume within physiological limits, matching output to venous return. Now we got to pay attention to these exam traps. Examiners exploit the mathematical relationship between heart rate, filling time, and volumetric output. A favorite trap is designing a question where a drastically elevated heart rate is assumed to continuously increase cardiac output. Remember the strict physical constraint highlighted on slide 5. That means this slide. That is, a very high heart rate severely shortens diastolic filling time, which causes a lower end diastolic volume, a lower stroke volume, and it can ultimately cause overall cardiac output to fall. Another classic trap involves the conceptual definition of the ejection fraction itself. Students often incorrectly assumes that percentage calculated by ejection fraction formula represents the total percentage of blood fully ejected from the entire body's reservoir. Pay close attention here. The ejection fraction refers strictly to the performance of single ventricle per single heartbeat, not a systemic body-wide percentage. To master performance shifts on high-yield exam questions, remember that during healthy physiological exercise, heart rate and stroke volume both increase simultaneously, allowing cardiac output to reach an impressive 20 to 25 liter per minute in trained athletes. To link this slide back to our broader circulatory system layout, remember that because the pulmonary and systemic circuits are arranged in series, the stroke volume and total cardiac output values calculated here must remain identical for both the right and left ventricles to avoid fatal fluid accumulation. And that is discussed under this title of the volumetric rule of continuity. To understand how the heart coordinates its own rhythm, let us build a map of the conduction system using fundamental principle. The heart is a myogenic pump, meaning its contraction is not initiated by the central nervous system, but rather by specialized modified cardiomyocytes that have traded their contractile machinery for electrical propagation features. To maximize the volumetric efficiency of this pump, the electrical signal must follow a strict, highly organized sequential pathway so that the muscle tissue depolarizes in a precise wave-like motion. Let us position our initial electrical power source on the conceptual map. The sinoatrial node or SA node, located high within the posterior wall of the right atrium, pointed by laser over here. This group of cells exhibits the fastest rate of spontaneous diastolic depolarization. We will learn about depolarization in muscle metabolism lecture later. Let us position our initial electrical power source on the conceptual map. The sinoatrial node or SA node, located high within the posterior wall of the right atrium, pointed by laser over here. This group of cells exhibits the fastest rate of spontaneous diastolic depolarization, or in other words, this SA node is the region where the electrical activity initiates very first. This intrinsic property of the SA node allows it to get activated before any other region, establishing the baseline rhythm of the heart. The localized action potential or impulse generated here rapidly propagates through the surrounding atrial syncytium. The word syncytium here simply means that the heart cells or cardiac myocytes are in an arranged order, and all the cells of the heart of this region get activated in symphony or are synchronized together. So as we were saying, the impulse generated here propagates through the surrounding atrial syncytium, indicated by these white arrows. The widespread depolarization or activation causes both atria, that means left and right atria, to contract in unison. Now let us introduce an essential structural boundary. If we draw the atria, we need to understand that the atria are completely separated from the ventricles by a non-conductive fibrous skeleton. That means if the signal wants to get to the next regions, it must follow a single specific gateway. And let us place that gateway at the atrioventricular junction, indicated by the pointer. Atrioventricular junction starts from atrioventricular node, this place, and it extends if we follow the yellow line up until here. So this is the AV junction, and the first part of the junction is AV node, and our point of interest right now is this AV node or atrioventricular node. As we were saying, the impulse was first generated in the sinoatrial node. As the impulse enters the AV node, it encounters a region of significantly high electrical resistance due to smaller cell diameters and fewer junctions. This architectural design creates a critical operational pause. That means the impulse doesn't get propagated from SA node to AV node immediately. There's a slight delay in between the propagation of the signal. This temporary delay ensures that atrial mechanical contraction is fully complete, permitting the atria to empty their blood volume into the lower chambers before the powerful ventricles receive the signal to contract. Let us track the impulse as it exits this delay gate. We mean the signal has already come to AV node. Now the signal will propagate further. The signal is channeled into a specialized fast conducting bundle located within the upper interventricular septum. That means if this is the septum, the atrioventricular bundle or bundle of Hees, which is universally known by this name, this bundle is fast conducting. And this bundle, starting from here, quickly splits into two divergent pathways, one going this direction, another one going to the right side, which we can tell as right and left bundle branches that raise down either side of the septum toward the apex of the heart. This is the apex of the heart. Finally, at the apex, these branches curve upward like this, and in case of left ventricle, like this. After curving upward, these branches ramify into a vast subendocardial network of massive rapid conduction cells. You can see over here this, and all these as well. And they are called the Purkinje fibers. These fibers possess an incredibly fast conduction velocity, allowing them to instantly distribute the propagation wave throughout the thick ventricular wall. This is the whole ventricular wall we're talking about, and this is pretty thick. And we know why it is thick. Because these fibers terminate near the apex and spread upward toward the base. This region is the base of the heart. As this Purkinje fiber terminates toward the base of the heart, the ventricles contract from the bottom up. That means the contraction starts here and it propagates toward the base. To make it more precise, this contraction style creates upward ringing motion, which efficiently drives blood toward the high pressure semilunar exits. One semilunar exit is here, another one is here. Having mapped this electrical sequence from first principles, let us now ground these concepts in the exact steps, values, and diagnostic traps on slide 6. As outlined on slide 6, the heart relies on an intrinsic electrical system that coordinates the mandatory contraction sequence from atria down to the ventricles. The mechanism starts with sinoatrial node or SA node, which is located in the right atrial wall. This SA node functions as the primary pacemaker of the heart, generating a resting rate of 60 to 100 beats per minute, which causes both atria to contract at the same time. Then comes AV node, which is positioned at the AV junction. This node imposes a crucial delay of approximately 0.12 seconds, functioning as a backup pacemaker, firing at 40 to 60 beats per minute if the primary SA node fails. Then comes the bundle of Hees. The electrical signal moves from the AV node into the bundle of Hees, which quickly splits into the left and right bundle branches along the interventricular septum. Finally comes the Purkinje fibers. These fibers are distributed throughout the ventricular walls, and they provide the absolute fastest conduction velocity in the entire heart, driving a highly coordinated contraction that sweeps from the apex up toward the base. From our discussion now, we can confidently say that SA node is the primary pacemaker of the heart, and AV node has slow conduction velocity, which causes delay, and it is a functional design. And if SA node fails, then AV node becomes the backup pacemaker, which generates 40 to 60 beats per minute in case of SA node failure. And Purkinje fiber have the fastest conduction velocity in heart compared to all other conduction tissues. All of them are high yield information that you need to inscribe in your mind. A few more nuggets that I would like to share is that the AV nodal pause or delay is 0.12 second, and this ensures complete atrial emptying before ventricular contraction can initiate. Now listen to these high yield exam traps to protect your score, as examiners frequently use electrocardiogram wave configurations to test your comprehension of this conducting sequence. A classic trap is asking you to point out the specific wave where atrial repolarization occurs. When the massive ventricular walls are depolarizing, that means atrial repolarization or relaxation occurs when the ventricular walls are preparing to get contracted. To get a clear grasp of what I'm saying, let's look at this ECG graph and listen to the interpretation. This image shows a standard electrocardiogram waveform diagram, mapping the heart's electrical activity during one cardiac cycle. It outlines the specific waves, segments, and intervals recorded during a heartbeat. Let us discuss the main waveforms. First comes the P wave. P waves represent atrial contraction through depolarization. You might get confused listening to the word depolarization, but you can just understand depolarization means the atria are starting to contract. So simply, this P wave means atrial contraction through depolarization. Then comes the QRX complex. This signals ventricular depolarization and contraction. Look how huge this looks on the graph. Then comes Q wave. This Q wave shows electricity crossing the middle wall. That means interventricular septum. The R wave shows the main surge squeezing the chambers to pump blood. And the S wave shows the signal finishing up at the top outer walls. Then this T wave will represent ventricular recovery and repolarization. The word recovery usually means the ventricles are relaxing after a massive contraction. Now we can discuss the intervals and segments. First comes the PR segment. This PR segment measures the conduction delay at the atrioventricular node. The ST segment is a flat baseline where ventricles finish contracting. The PR interval is the time from atrial depolarization to ventricular depolarization. Don't get confused with the word depolarization. Depolarization means the ventricles are recharging for a contraction. The QT interval is the total ventricular contraction and reset time. Remember, after the ventricles contract, they need a time to reset again. You can exchange the word reset with repolarization to have a better understanding. Now that we know the basics of ECG, we can understand the following sentence I am about to mention. You have to remember atrial relaxation or repolarization occurs precisely when massive ventricular walls are depolarizing. That means atrial repolarization and ventricular depolarization happen at the same time. This atrial repolarization event is completely hidden by the dominant QRX complex, which signifies ventricular depolarization. Why does it happen? Because the ventricles are massive compared to the thin wall atria, and the electrical signals generated by ventricular depolarization is much larger than the atrial repolarization. Now we can fully understand why it is written that atrial repolarization is hidden by QRX complex on ECG. Another notorious trap involves analyzing conduction delays. Examiners will present a question showing a prolonged PR interval. That means this interval will increase. And showing that event, examiners will try to trap you into classifying it as a complete heart block. Always remember, a prolonged PR interval strictly defines a first-degree heart block, which represents slow or impaired AV conduction. AV means atrioventricular. It doesn't mean a complete or total conduction block. To seamlessly correlate this conduction map with diagnostic waves, memorize this high-yield rule that we have discussed previously. The P wave represents atrial depolarization. The QRX complex signifies ventricular depolarization, and the T wave marks ventricular repolarization. On complex clinical questions, remember that the total length of QT interval serves as an accurate proxy for the overall duration of the mechanical ventricular systole. We established at the start that there are two circuits in series. Now let us compare them properly, because the IMAT tests this comparison directly and the differences are stark. And that means now we will study the pulmonary versus systemic circulation. Let me repeat it. The human cardiovascular plumbing plan is organized as a double circulation system, where the circuits are arranged strictly in series. This in-series configuration enforces a fundamental rule of fluid dynamics. All blood must complete both loops sequentially. If you trace a single erythrocyte, it cannot bypass one loop to go to the other. It is physically bound to complete a full sequence through both networks to maintain homeostatic balance. That means if this erythrocyte starts its journey in the left ventricle, it will first go to the body, then it will come again to the right atrium of heart. From right atrium, it will go to the lungs, and from lungs, it will come to the left atrium. And obviously, from left atrium, it will go to the left ventricle, which completes the full journey of erythrocyte in a series. Let us position the first loop on our conceptual map. The pulmonary circulation. The circuit fulfills a singular homeostatic purpose, and that is gas exchange at the respiratory membrane. It is driven by the right ventricle, which pumps spent blood out into the pulmonary trunk. Pulmonary trunk will branch out to give off pulmonary artery. Because the distance from the heart to the lungs is structurally short and the pulmonary capillaries form a massive, highly compliant vascular bed shown over here, the pressure required to drive this circuit is remarkably low. The arteries we were talking about, that is pulmonary artery, carry deoxygenated blood. That's why color-coded blue over here. And they carry the blood away from the heart towards the lung, where carbon dioxide is unloaded and oxygen is picked up. Once it is oxygenated, the blood travels through the pulmonary veins directly to left atrium. Now let us introduce the second loop, which is arranged in series right after the first. We mean the systemic circulation. This vast network is built to deliver oxygen and nutrients to every peripheral organ bed, spanning from the upper body to the lower body, including specialized paths like the renal circulation to the kidneys and circulation to the gastrointestinal tract. Because it must overcome the immense friction and resistance of all combined systemic tissues, this circuit requires a much higher pressure head. It is driven by the powerful left ventricle, which ejects oxygenated fluid into the aorta. In this systemic circuit, the arteries carry highly oxygenated blood out to the organs, where exchange capillaries distribute the gas. As depicted on this image, the gastrointestinal tract incorporates a specialized inline loop, the hepatic portal vein. This one is the hepatic portal vein. This hepatic portal vein routes nutrient-rich blood to the liver before it enters the general venous pooling. Finally, the spent deoxygenated blood is collected by the vena cava, depicted over here, which returns to the right atrium, completing the master cycle. Now that we have mapped out the anatomical and pressure relationship of these dual circuits from first principles, let us transition directly to our text and anchor these concepts to the exact parameters, comparisons, and high-yield traps on slide 7. As defined on slide 7, the human body utilizes two distinct circuits arranged in series, ensuring that all blood completes both circuits in a mandatory continuous sequence. The pulmonary circulation is structurally driven by the right ventricle and operates under a low pressure head of approximately 25 by 10 mm of mercury. Conversely, the systemic circulation is driven by the powerful left ventricle and demands a high homeostatic pressure of approximately 120 by 80 mm of mercury. Within the pulmonary circuit, the arteries carry deoxygenated blood to the respiratory organs, while the veins carry oxygenated blood back to its destination in the left atrium. And within the systemic circuit, the arteries carry oxygenated blood out to tissue beds like the kidneys and liver, while the veins carry deoxygenated blood back to its destination in the right atrium. And as usual, pay critical attention to these high-yield exam traps to protect your score. A standard trap is designing a question that states that all arteries carry oxygenated blood. Remember the explicit rule highlighted on slide 7. Pulmonary arteries carry deoxygenated blood, while pulmonary veins carry oxygenated blood, entirely reversing the classic systemic pattern. Another frequent trap relates the pressure differences of the circuits directly back to cardiac histology. Examiners will ask you to identify the mechanical reason behind the structural asymmetry of the ventricular walls. Remember the trap. It is the lower pulmonary pressure requirement of 25 by 10 mm of mercury that perfectly explains why the right ventricular wall is significantly thinner than the left ventricular wall, which must fight high systemic resistance. To bridge fluid physics with clinical reality on your exam, remember that because these two loops are in series, an equal cardiac output from both ventricles is absolutely mandatory, meaning they share the exact same volumetric flow rate. Now let us come to our next topic, which is coronary circulation and the portal system. To master the specialized local networks of the human circulatory architecture, we will establish a blank conceptual map detailing the coronary and hepatic portal circuits based entirely on first principle. While the heart spends its entire day generating pressure to propel blood out to the systemic body, the myocardium itself is a highly metabolic muscular tissue that cannot rely on the blood sitting inside its hollow chambers for nutrient exchange. The walls are far too thick for efficient diffusion. Therefore, we must introduce a dedicated structural system to nourish the engine itself, which brings us to the coronary circulation. Let us position the origins of this circuit on our conceptual map. Right at the base of the ascending aorta, that is over here, immediately superior to the aortic semilunar valve, the valves are here, the aortic wall dilates into three small pockets called the aortic sinuses. As it is a 2D section, we only see one sinus, but a 3D picture would have shown three sinuses together over here. And we anchor our primary supply channels, that means the coronary arteries, in this sinus area. Branching out from these sinuses, we find two main paths: the left main coronary artery and the right main coronary artery, as clearly mapped on this specific image. Now let us trace their structural pathways. The left coronary artery travels a short distance before bifurcating into two massive regional conduits: the left anterior descending coronary artery, as pointed here, and circumflex coronary artery, which sweeps around the left side of the heart. Meanwhile, the right coronary artery tracks along the opposite side, wrapping around to supply blood to the right ventricle and the posterior walls of the left ventricle. Now let us introduce a remarkable hemodynamic paradox that governs how fluid moves through these coronary conduits. In any other organ bed in the human body, tissue perfusion peaks during ventricular contraction or systole, when arterial pressure is highest. But in the heart, the exact opposite occurs. When the myocardium contracts violently during systole, the massive intramyocardial compression physically squeezes the small coronary vessels shut. That means, due to the massive contraction of the ventricles, these coronary arteries would be squeezed, which will halt the forward flow. Therefore, coronary blood flow is almost entirely diastolic. That means the blood flow will be present when the chambers are relaxing. Now let us transition our conceptual map from the local engine supply to a unique structural bypass system within the abdominal cavity. That is the hepatic portal system. In a standard circulatory sequence, blood travels from an artery to a single capillary bed for exchange, which then drains into a vein straight back to the general venous pool. A portal system, on the other hand, breaks this rule by placing two distinct capillary beds in series, linked by a portal vessel, which you can see in this diagram. You see there's first capillary bed and this is the second capillary bed. Both of the capillary beds are connected by hepatic portal vein. Let us position this inline sequence to understand its metabolic why. You see the blood is moving through the digestive organs, such as stomach, intestines, or spleens. During its journey, the blood picks up reabsorbed nutrients alongside potentially harmful ingested toxins. If this blood drained immediately back into the inferior vena cava, that means in this vessel, traveling directly from this path, or let's say this path, these raw molecules would be distributed to the brain and body unfiltered. To prevent this, we introduce a massive venous highway, the portal vein here. This vessel collects all nutrient-rich blood from the gut capillaries and channels it directly into a second downstream capillary network located inside the liver. This capillary bed is actually inside the liver. And in this place, in this capillary bed of liver, the cells of liver store and detoxify the blood. Only after filtering through this second liver bed, the blood collect into the hepatic veins here, which finally empty into the inferior vena cava reservoir to rejoin general circulation, which eventually would go to the right atrium. Now that we have mapped out the mechanical gating of coronary flow and the sequential plumbing of the abdominal portal network, let us transition directly to our text and anchor this concept. To formalize the features on slide 8, the coronary arteries arise directly from the aortic sinuses just above the leaflets of the aortic valve. The left coronary artery structurally branches into the left anterior descending coronary artery and the circumflex coronary artery to supply the anterior and lateral regions of the heart. The right coronary artery wraps around the cardiac skeleton to distribute blood directly to the right ventricle and posterior aspects of the left ventricle. A crucial mechanical feature dictates that coronary flow is strictly diastolic, occurring because the myocardium is relaxed and the local exchange vessels are no longer compressed by contracting muscle fibers. Hepatic portal system is specialized macro circuit which functions as a strict inline sequence. In this system, blood travels from gut capillaries into the portal vein and moves into liver capillary bed, which drains into hepatic veins and finally enters the inferior vena cava. Now coming to the exam traps. A favorite trap is asking a question that implies the coronary arteries fill during peak ventricular ejection or systole because they branch off the high pressure aorta. Remember the absolute rule highlighted on this slide. Coronary arteries fill during diastole, not systole, making them highly sensitive to changes in diastolic relaxation time. Another classic trap involves the directionality of the portal blood flow. Examiners will try to confuse your terminology by writing multiple choice questions stating that the portal vein drains blood away from the liver toward the intestinal tract. Keep this sequence perfectly straight in your mind. The portal vein carries blood to the liver from the gut, acting as an inbound processing line, not an outbound drainage pathway. To bridge this vascular pathway with high-yield clinical pathologies on your exam, remember that any structural blockage or cirrhosis in the liver capillary bed backs up pressure into the portal system, causing portal hypertension, which classically presents with esophageal varices. That means the veins of the esophagus will become swelled, and ascites, which means accumulation of fluid in the abdominal cavity, and splenomegaly, that means the enlargement of spleen. On complex biochemical absorption questions, remember that dietary lipids serve as a fascinating structural exception to this portal highway. Instead of entering the portal vein, fat molecules are packaged into chylomicrons, and they completely bypass the portal system by traveling through intestinal lacteals, then into thoracic duct, and directly into the systemic subclavian vein. More on lacteals on the digestion chapter later. Let's map out blood vessel structures from scratch to see how the circulatory system functions. The vascular system is not a set of inert plumbing pipes. It is a highly specialized, layered series of living tubes designed to withstand immense pressure variation, regulate regional distribution, and permit molecular transport. To understand how these tasks are divided, we must focus on the microscopic architecture of the vessel wall, which classically features a three-layered configuration. I have given two images here to have a clear understanding. You see layer number 1, 2, and 3. And in this image, we have artery and vein structure. Similarly, in artery, in this image, three layers are seen. Now let us position the innermost layer on our conceptual map, which is tunica intima. In this specific diagram, tunica intima is this portion, and in this diagram, this is tunica intima. This tunica intima layer is composed of a single continuous sheet of simple squamous endothelial cells lining the lumen, sitting flush against the flowing blood. This endothelium provides an ultra-smooth, non-thrombogenic boundary that minimizes fluid friction. Moving outward, we establish the intermediate layer, the tunica media. This layer consists of smooth muscle cells interspersed with elastic fibers. The relative abundance of these two components, smooth muscle cell and elastic fibers, dictates a vessel's compliance and vasoactive capabilities. Finally, we place the outermost anchoring coat, the tunica adventitia or tunica externa, a tough sheet of dense connective tissue packed with collagen fibers that structurally reinforce the vessel and prevent mechanical overstretching. Now let us look at how this basic structural blueprint adapts to specialized functional roles as blood travels downstream from the heart. To have a perspective, let's draw the heart over here, and this is our left ventricle. The largest conduits, or the arteries that exit the left ventricle, are elastic arteries, and in this case, aorta leaves left ventricle. So this is elastic artery. This elastic artery possesses an exceptionally thick tunica media rich in elastic fibers, as illustrated in this segment. The structural density allows them to act as a pressure reservoir. They distend during peak systole to store potential energy and passively snap back during diastole, utilizing elastic recoil to buffer pressure swings and conduct a smooth, continuous pulsation. Further down the line, this transition into muscular artery, where the tunica media shifts to a predominantly smooth muscle architecture. These vessels are built to distribute blood to specific organs, adjusting their luminal diameters to govern regional blood flow. As we move closer to microcirculation, the tubes taper into arterioles. That means we started with aortas like this, or elastic arteries, then it became muscular artery, and eventually it turns to small arterioles. And we will talk about arteriole right now. And these arterioles possess a thin adventitia, or tunica externa, and a wall heavily dominated by smooth muscle relative to their small size, as you can see in tunica media. The high proportion of smooth muscle turns them into the main resistance vessels of the body, allowing them to contract or dilate to control capillary flow and regulate overall peripheral vascular resistance. Now let us remove these outer structural layers entirely to arrive at the site of ultimate physiological utility. That means we will study capillaries right now. As visible on this image, a capillary, as you can see here, this capillary strips away tunica media and adventitia, leaving nothing but a single delicate layer of endothelium. Being only one cell thick, this minimalist barrier minimizes the physical distance over which diffusion must occur, serving as the primary site of exchange for oxygen, carbon dioxide, nutrients, and waste transfer. After exiting this exchange bed that we call capillary bed, this is the capillary bed in the image. After exiting this capillary bed, the vessels merge into thin-walled veins. First, you see venule here. These venules will become veins and return the components to the heart again. This is a microscopic image of artery and vein, and you see veins have a large, highly compliant lumen, and veins also feature valves to guarantee unidirectional return, functioning as a high-capacity blood reservoir. That means veins store blood. Now that we have mapped out the layered histology and downstream transitions of the vascular tree from first principles, let us transition directly to our text and anchor these concepts to the exact definitions, features, and high-yield traps on slide 9. To formalize the framework from slide 9, standard blood vessels are structurally composed of three distinct tunica layers. The innermost tunica intima, made of endothelium, the middle tunica media, composed of smooth muscle and elastic fibers, and the outermost tunica adventitia or externa consisting of collagen. And capillaries stand as a structural exception, composed entirely of an endothelial layer with no tunica media and no adventitia present. Now our table starts with elastic artery. This artery is characterized by a thick elastic wall, and these vessels utilize elastic recoil to buffer sharp pressure fluctuation. That means it can buffer pressure, which functions to conduct blood and smooth out harsh cardiac pulsations. Muscular arteries feature a thick muscular wall specialized to distribute blood directly to target organs and regulate regional blood flow. Arterioles combine a thin wall with smooth muscle to act as the primary resistance vessel of the human body, serving to control capillary flow and regulate peripheral vascular resistance. Capillary is built just one cell thick, and they act as the exclusive site of metabolic exchange, facilitating the cross-membrane transfer of oxygen, carbon dioxide, nutrients, and waste products. Veins, finally, are characterized by thin walls, large internal lumen, and specialized venous valves designed to return blood to the heart and serve as a massive blood reservoir. Now pay critical attention to these high-yield exam traps. As usual, a major trap involves identifying which vessels have the highest concentration of smooth muscle relative to their total scale. Students often guess large muscular arteries because of their name, but the trap highlighted on this slide is that arterioles possess the thickest smooth muscle relative to their diameter, making them the primary regulators of total peripheral vascular resistance. Another classic multiple choice trap targets the minimalist histology of the exchange beds. Question stems will try to trick you by asking about the thickness or composition of the smooth muscle or collagen coats inside a capillary wall. Remember the strict rule from this slide. Capillaries are purely one cell thick and possess absolutely no tunica media and no tunica adventitia. To master volume distribution matrix on your exam, remember that venous system functions as the body's primary capacitance network, with approximately 60 to 70% of total blood volume residing passively in veins at rest. That means veins work as reservoir. To bridge this structural layout with fluid physics, finally, we need to remember that arterioles are the primary PVR vessels. PVR stands for peripheral vascular resistance, and if any minor adjustment is done to the smooth muscle tone of these arterioles, it will trigger exponential change in resistance, as fluid resistance is inversely proportional to the fourth power of the vessel radius. That means capital R is inversely proportional to 1 over R to the power 4. This small r stands for vessel radius, and this capital R stands for fluid resistance. Now we will master the physiological control of perfusion. Imagine this is a blood vessel or conduit. If we consider blood pressure, then blood pressure is the hydromechanical force extracted by circulating blood against the structural walls of the conduit system. Mathematically, this system is governed by a fundamental product equation. Blood pressure equals the total volume of fluid pumped per minute multiplied by total frictional resistance, which is encountered across the microcirculation. To systematically analyze this, we define two primary pressure peaks that occur within a single cardiac timeline. First, let us talk about systolic pressure. This is the absolute peak force attained as left ventricle propels its stroke volume into arteries. And then comes diastolic pressure. This is the lowest baseline force resting within the conduits as the heart undergoes diastolic relaxation. To calculate the true perfusion pressure experienced by peripheral organs over time, simple averaging is hydrodynamically inaccurate because the heart spends twice as long in a state of relaxation as it does in a state of contraction. Therefore, we introduce the concept of pulse pressure. which represents the mathematical difference between our systolic and diastolic peaks. By taking the resting diastolic pressure and adding exactly 1/3 of the pulse pressure, we derive the true mathematical mean systolic tissue perfusion, a crucial index known as the mean arterial pressure. And already we have said the pulse pressure is the difference between the systolic peak and the diastolic peak. We can write pulse pressure as systolic blood pressure minus diastolic blood pressure for simplicity. Now let us introduce the neural and endocrine feedback loop to stabilize this mean pressure around a strict homeostatic set point. We will talk about homeostasis later in any other lecture, but for now just understand that homeostasis means the tendency of the body to keep all the metrics under balance. The body separates the defensive responses into a rapid millisecond neural reflex and a slow long-term hormonal cascade. Let us position the neural mechanism on our map first, the baroreceptor reflex. Now let us position the neural mechanism first, which is the baroreceptor reflex. These specialized receptors are embedded within the carotid sinus and the aortic arch of our body. For your information, carotid sinus is a slightly dilated area, as you can see here, at the base of the internal carotid artery, which is located in the neck. Now we are ready to go to the mechanism. When the blood pressure goes up, the blood vessel walls stretch, and this stretching immediately sends faster electrical signals through neck nerve, which are carotid nerve 9 and 10, to the control center of the brain. 9th cranial nerve is known as glossopharyngeal nerve, and 10th cranial nerve is called the vagus nerve. They send the signal to the nucleus tectus solitarius of the medulla. Then NTS processes this sensory input and alters the autonomic balance. Autonomic means we don't control it, our body automatically controls it. So NTS further activates another nucleus inside the brain called nucleus ambiguus to boost parasympathetic output via the vagus nerve. You just need to remember parasympathetic tries to put our body in the state of rest, and sympathetic nervous system prepares our body for emergency. So to complete the sentence, we would say again that NTS processes this sensory input. It activates the nucleus ambiguus to boost parasympathetic output via vagus nerve, which goes directly to the sinoatrial node. The nucleus tectus solitarius also sends a signal to caudal ventrolateral medulla to suppress rostral ventrolateral medulla, which crushes the sympathetic outflow through the intermediolateral column. Hold on, you don't need to be afraid and you don't need to remember all these names. I'm just saying those names to give a clear map of the mechanism. What you need to remember, I have put that in the anchor slides. So when we would review the anchor slides, you would easily find out what to know and what to skip. As we were saying, when this intermediolateral column gets the signal, it crushes the sympathetic outflow. And as a result, this sudden shift forces heart rate, stroke volume, and peripheral vascular resistance to fall, rapidly pulling the pressure spike back down to baseline. So simply put, we can say baroreceptor detect the high pressure and normalize it with the neural mechanism, which acts on the blood vessels and heart itself. Finally, let us introduce the slow endocrine network designed to manage sustained pressure deficits, which is called the Renin-Angiotensin-Aldosterone system, or simply RAAS. As illustrated on this image, when systemic blood pressure drops, the specialized macula densa cells inside the renal nephron sense the drop in fluid flow or sodium concentration. This triggers the neighboring juxtaglomerular cells to secrete an enzyme called renin directly into the bloodstream. At the same time, the liver continuously synthesizes and releases a massive plasma protein known as angiotensin. Once renin enters the blood, it acts as an enzyme catalyst, breaking down angiotensinogen to produce an inactive intermediate called angiotensin 1. To activate this molecule, we must introduce a key enzyme, as angiotensin 1 circulates through the vast network, or I should say vast microvascular network of the lungs, it encounters angiotensin converting enzyme or ACE bound tightly to the pulmonary endothelial membrane. ACE cleaves two amino acids from the chain, converting this molecule of angiotensin 1 into a highly potent hormone which is called angiotensin 2. Angiotensin 2 immediately executes a dual defensive strategy to raise pressure. First, it binds directly to vascular smooth muscle, shown over here, to cause widespread vasoconstriction to elevate resistance, while simultaneously stimulating the outer cortex of the adrenal gland to secrete aldosterone. Aldosterone travels to the renal distal tubules and collecting ducts, upregulating apical sodium channels to maximize sodium uptake, which pulls water along with it. This combined fluid retention expands total blood volume, permanently restoring long-term blood pressure balance. So once again, we can say that high BP will activate the macula densa cells, and it will further activate juxtaglomerular cells, which will secrete renin, and renin will break down angiotensinogen to angiotensin 1. And angiotensin 1 will be converted to angiotensin 2 by the enzyme called ACE. This angiotensin 2 will cause vasoconstriction. That means the blood vessels will be constricted, which will increase blood pressure. And at the same time, angiotensin 2 will tell adrenal cortex to secrete aldosterone. And this aldosterone will tell the tubules of kidney to take up as much sodium as it can, which will eventually pull water along with it, which will increase the volume inside the blood vessels, and eventually it will increase blood pressure to normalize the system. ADH or antidiuretic hormone also increases blood pressure, but this is not the part of RAAS or Renin-Angiotensin-Aldosterone system. So we will not study about it here. We will study in depth about ADH in our renal system lecture. Now that we have mapped out these mathematical parameters, neural ergs, and enzymatic cascades from first principles, let us transition directly to our text and anchor these concepts. Formally mapping out slide 10, systemic blood pressure is strictly defined by the mathematical product of cardiac output and peripheral vascular resistance, and systolic blood pressure represents the peak pressure generated during ventricular ejection, whereas diastolic blood pressure marks the baseline pressure remaining during ventricular relaxation. Mean arterial pressure is equal to diastolic blood pressure plus 1/3 of pulse pressure, and pulse pressure is measured through this explicit formula, which is pulse pressure is equal to systolic blood pressure minus diastolic blood pressure. Now going into the mechanism segment, where it starts with fast baroreceptor reflex. This rapid neural loop monitors stretch via the mechanoreceptors located in the carotid sinus and aortic arch, routing signals to the medulla to adjust autonomic output. The mechanism goes as BP when is high, baroreceptor increases the firing, which in turn increases parasympathetic outflow and decreases sympathetic outflow, which causes the heart rate, stroke volume, and peripheral vascular resistance to fall. Then comes the slow endocrine cascade. The Renin-Angiotensin-Aldosterone system, or RAAS, regulates chronic blood pressure drops by initiating an enzymatic sequence that culminates in widespread vasoconstriction and sodium or water retention. To formalize it, let's also look at the intermediate sequence. When blood pressure falls, that means low BP, kidney secretes renin, which breaks angiotensinogen and makes angiotensin 1. This angiotensin 1 is converted to angiotensin 2 by an enzyme called ACE, secreted by lungs. This angiotensin 2 causes vasoconstriction, and at the same time, it tells the kidney to secrete aldosterone. To be more precise, aldosterone is secreted by the adrenal gland, which is situated over kidney. And finally, it causes sodium and water retention. Now, pay critical attention to these exam traps. The notorious trap is asking you to locate the tissue source of angiotensin converting enzyme, or ACE. Many students guess the kidney as source of ACE, but remember the explicit anatomical trap. ACE is located within the pulmonary endothelium of the lungs, not the kidney. Another common trap focuses on the exact enzymatic function of the renin. Multiple choice questions will frequently state that renin directly produces angiotensin II or directly cleaves angiotensinogen into angiotensin II. Pay strict attention here. Renin acts on angiotensinogen to produce angiotensin I, not directly angiotensin II. To bridge this endocrine cascade with high-yield clinical pharmacology, remember that ACE inhibitors, a type of medication, function by blocking the conversion of angiotensin I to angiotensin II, effectively reducing peripheral vascular resistance, blunting aldosterone secretion, and decreasing total blood volume to treat hypertension. On primary physiology questions, always remember that baroreceptors do not directly measure fluid dynamics. They respond exclusively to mechanical stretch caused by the pressure variations, and their sensory bodies are located explicitly within the vessel walls. Now let us turn to the blood itself, starting with the most abundant cells in the body, the erythrocytes. Then we will talk about hemoglobin as well. We will establish a conceptual map detailing the physiology of erythrocytes and hemoglobin based entirely on first principles. The primary task of blood gas transport requires a highly optimized container capable of navigating microscopic capillary beds while maximizing gas packing efficiency. To achieve this, we introduce the specialized red blood cells. The erythrocyte features a unique biconcave disc geometry. If you draw an erythrocyte, you will see it is concave above and beneath as well. This geometric arrangement provides an exceptionally high surface area to volume ratio, which drastically accelerates the diffusion rate of gases across the cell membrane. Measuring just 7 to 8 micrometers in diameter, these cells have a normal concentration range of approximately 4.5 to 5.5 million cells per microliter of blood. To maximize internal storage capacity for its gas-carrying cargo, a mature red blood cell undergoes a profound developmental sacrifice. It completely ejects its nucleus, mitochondria, and ribosomes. Because it lacks mitochondria, the mature erythrocyte cannot execute oxidative phosphorylation. Instead, it metabolically is restricted to passive anaerobic glycolysis for its basic ATP requirements. This survival strategy guarantees a fixed cellular lifespan of approximately 120 days, after which the aged, rigid membranes are broken down by macrophages in the spleen and liver. Now let us place the master transport protein inside this biconcave envelope, which is hemoglobin. A single hemoglobin molecule is a quaternary tetramer built from a precise layout. 4 globin polypeptide chains consisting of 2 alpha chains and 2 beta chains. If we count, this is 1, 2, 3, and 4. In total of 4 chains. And out of 4 chains, we will have 2 alpha, 1 over here, another 1 over here, and 2 beta, 1 over here, another 1 here. Embedded symmetrically within each of these 4 globin chains is a non-protein prosthetic group known as a heme group, indicated here with a black big dot. At the absolute geometric core of each heme group sits a single iron atom in its reduced ferrous state, designated as Fe2+, because each ferrous iron atom can form a single reversible coordinate bond with the diatomic oxygen molecule. One fully packed hemoglobin molecule can carry a maximum of 4 oxygen molecules. When oxygen binds to its first subunit, it induces a conformational shift that is transmitted to the remaining chains, a mechanical phenomenon known as cooperative bonding. This nonlinear cooperative interaction alters gas affinity dynamically, yielding a characteristic sigmoidal oxygen hemoglobin dissociation curve that we have seen in the respiratory system lecture. Now finally, let us remind ourselves of the transport challenge, which is moving carbon dioxide waste away from the tissues. The body coordinates this transport through 3 distinct parallel mechanisms. A minor fraction of approximately 7% remains simply dissolved directly within the liquid plasma. Another 23% enters the erythrocyte and binds chemically to globin amino terminals of the protein itself, forming carbaminohemoglobin. The vast majority, however, undergoes a chemical transformation. Approximately 70% of all carbon dioxide is converted inside the cell into highly soluble bicarbonate ions, which are pumped out to travel passively in the plasma, acting as the primary buffer system of human blood. Now that we have mapped out these cellular dimensions, structural tetramers, and gas fractions from first principles, let us transition directly to our text and anchor these concepts to the exact definitions, features, and high-yield traps on slide 11. First things first, red blood cell is a biconcave disc measuring 7 to 8 micrometers with a standard resting concentration of 4.5 to 5.5 million per microliter. A mature RBC possesses no nucleus, no mitochondria, and no ribosomes, relying exclusively on anaerobic glycolysis as its metabolic energy source, with an average lifespan of 120 days before destruction in the spleen and liver. Remember, RBC can perform aerobic glycolysis. It relies entirely on anaerobic glycolysis. As seen in our conceptual mapping, each hemoglobin molecule is composed of 4 globin chains, 4 heme groups, and 4 iron ions. To be more precise, ferrous ion, allowing one hemoglobin molecule to carry a maximum of 4 oxygen molecules. The structural layout of hemoglobin enforces cooperative binding, which directly gives rise to the classic sigmoidal oxygen-hemoglobin dissociation curve. Our slide also breaks down carbon dioxide transport into 3 explicit pools: 7% dissolved in plasma, 23% bound to hemoglobin as carbaminohemoglobin, and 70% converted into bicarbonate in plasma. If we want to remind ourselves of the mechanism, then we can talk about oxygen capturing sequence. In the high oxygen environment of the lungs, oxygen diffuses into the biconcave cell, that means RBC, and binds reversibly to the internal ferrous ion centers, utilizing a cooperative chain reaction to rapidly maximize overall protein saturation. And at the tissue level, metabolic carbon dioxide waste diffuses into the bloodstream, where 70% is rapidly converted into soluble bicarbonate ions to be safely carried back to the lungs for respiratory elimination. Now coming to the exam traps, which will be directly asked in the questions. First favorite trap is asking whether a red blood cell consumes the oxygen it transports as it travels through systemic circuits. Remember the strict rule highlighted on this slide. Mature RBCs have no mitochondria and cannot perform aerobic respiration, a perfect physiological design trap ensuring they do not consume a single molecule of oxygen they carry. Another classic multiple choice trap targets the quantitative ranking of waste gas transport. Questions will try to trick you into stating that the main pool of carbon dioxide travels as dissolved gas or bound directly to hemoglobin. Pay strict attention here. Bicarbonate is the absolute most common form of carbon dioxide transport, accounting for a massive 70% of the total volume. Now coming to the exam pearls. On complex respiratory curves questions, always remember that sigmoidal curve mathematically dictates that hemoglobin's affinity for oxygen is not fixed. Its affinity increases progressively with each subsequent oxygen molecule bound to its iron core. In slide 12, we will talk about the soldiers of our immune system and also platelets, which help in blood coagulation. To master the dynamic cellular defense and repair networks circulating within our conduits, let us establish a blank conceptual map detailing the classification and function of leukocytes, also known as white blood cell, and platelets. While RBCs dominate gas transport, the fluid medium must also harbor highly specialized cellular components dedicated to active immune surveillance and mechanical hemostasis. To systematically organize these defense elements, we classify the nucleated leukocytes or white blood cells into 2 primary categories based on the microscopic presence or absence of prominent cytoplasmic storage vesicles. First would be granulocyte, simply meaning it will have grains inside the cytoplasm. Another one would be a granulocyte. Let us position our first primary family on the map. That means the granulocyte. The cells are visible on this image and are easily identified by their lobed nuclei and prominent granules. You see the nucleus inside the white blood cells has multiple lobes. The absolute vanguard of this group is the neutrophil, the most abundant white blood cell in human circulation. Packed with neutral staining vesicles, these cells function as rapid response phagocytes specialized in engulfing and destroying invading bacteria. Next, we place the eosinophils, which feature a distinct bilobed nucleus, lobe number 1 and lobe number 2. And it also has large, bright red granules, as you can see here. These specialized packages contain destructive enzymes optimized for anti-parasitic defense and the modulation of allergic inflammation. Completing the granulocyte trio are the basophils, which are heavily masked by massive deep blue-purple granules. Here, these dense storage vesicles are loaded with histamine and heparin, which are actively released upon stimulation to drive acute inflammation and vasodilation. Now let us introduce the opposite arm of the defense network, the agranulocytes, which lack conspicuous cytoplasmic staining. This family is divided into monocyte and lymphocytes. Monocytes are massive cells, characteristic kidney bean-shaped nucleus, as you can see here. It looks like a kidney. They are long-lived sentinels, and they circulate in an immature state before actively migrating across endothelial borders to differentiate into highly efficient tissue macrophages. As outlined on this image, these macrophages execute vital phagocytic digestion of large pathogen and cellular debris. In this image, you see the phagocytosis process of macrophage. The first one, where the antigens or pathogens are less than 10 micrometer in size, macrophages digest them. On the other hand, when the pathogen or foreign particles are greater than 10 micrometers or within the range of 10 to 100 micrometer, macrophage fuse to the foreign particle, then engulfs it and completes digestion. Next, we place lymphocytes over here. These are the central orchestrators of adaptive antigen-specific immunity. We will talk about immunity in a separate lecture. This family of lymphocytes splits into two main functional lineages. One is B lymphocyte, another one is T lymphocyte, which are structurally featured on our next images. First comes B cells. B cells are specialized to produce highly specific circulating Y-shaped proteins called antibodies, which drive humoral immunity to target extracellular pathogens. And T cell, which you see in this image, conversely coordinate cellular immunity. They divide into helper T cell lineages that manage the broader immune cascade and cytotoxic lineages that directly kill virally infected or mutated cells. Finally, let us introduce our primary physical repair element, the platelets. Platelets present as tiny irregular cytoplasmic fragments clustered together between the larger cells, as you can see here. They lack a nucleus because they are not independent cells at all. Rather, they are structural fragments that directly from the cytoplasm of massive parental cells located within the bone marrow. These fragments are packed with procoagulant proteins and signaling molecules, allowing them to rapidly adhere to exposed subendothelial collagen to form a localized plug that halts mechanical blood loss. Now that we have mapped out these cellular lineages, granular variations, and defense roles from first principle, let us transition directly to our text and anchor these concepts to the exact definitions, reference values, and high-yield traps. First comes the introduction of WBCs. White blood cells are fully nucleated cells with a standard healthy concentration range of 4,000 to 11,000 cells per microliter, operating primarily in immune defense. And platelets present as anucleated cell fragments with a normal concentration range established between 150,000 to 400,000 fragments per microliter. Now we have the subtypes in this table under the title Key Features. Neutrophil, eosinophil, basophils, all of them are granulocyte, and monocytes and lymphocytes are agranulocytes. Starting with granulocytes, neutrophils, they possess prominent granules and function as the most abundant white blood cell, specializing in the phagocytosis of bacteria. Eosinophils are characterized by distinctive red granules, serving a primary function in anti-parasitic response and allergy states. Basophils are white blood cells with heavy, large blue granules, acting as the primary agents for histamine release and acute inflammation. Let me introduce inflammation in a few words. Inflammation means the body is reacting to a trauma or injury to heal itself. Now coming to agranulocytes, starting with monocytes. Monocytes don't have any granules and they circulate in the blood before migrating to differentiate into highly active macrophages in peripheral tissues. B lymphocytes also lack granules and function as the primary drivers of humoral immunity by producing targeted antibodies. T lymphocytes also lack granules and drive cellular immunity, acting via helper or cytotoxic T-cell pathways. Now we need to pay attention to these exam traps stated here. The first trap is designing a question that lists platylate among a count of true independent cells or suggesting they can undergo active mitosis. Remember the explicit trap highlighted on this slide. Platylates are not true cells. They contain absolutely no nucleus and they're purely enucleate fragments shed from the cytoplasm of bone marrow megakaryocytes. Another classic multiple choice trap involves the precise biochemical difference between the liquid fraction of blood. Question stems will try to trick you into treating plasma and serum as completely identical terms. Memorize the absolute rule from slide 12. Serum is strictly defined as plasma minus its clotting factors, which occurs because fibrinogen and related proteins are completely consumed and removed during the formation of a physical blood clot. Now let me give you a few more nuggets of knowledge regarding leukocytes and platylate. To quickly solve differential white blood cell count questions on your exam, memorize the classic mnemonic, never let monkeys eat bananas, which perfectly ranks resting abundance from highest to lowest. Neutrophil, lymphocyte, monocytes, eosinophils, and basophils. And to bridge these microscopic components with downstream clinical mechanics, remember that while platylates form the immediate initial cellular plug to arrest bleeding, they must work in parallel with soluble plasma clotting factors to deposit a permanent fibrin meshwork to securely anchor the repair site. More on this in our next slide. Now we would master the dynamic physiological mechanisms that arrest bleeding. And for that, we would establish a blank conceptual map of hemostasis and coagulation based entirely on first principles. Let's say this is a vascular conduit, and if it is ruptured anywhere, the high hydrostatic pressure inside the vessel threatens to cause rapid fluid loss. That means the fluid will have the tendency to come out of the site of rupture. And to survive this mechanical insult, the body must instantly activate a localized, multi-phased biochemical defense system designed to minimize immediate flow, construct a temporary cellular seal, and deposit an immovable polymer network to permanently repair the breach. Let us position the initial immediate response on our conceptual map. Vascular spasm. The very millisecond a vessel wall sliced. Local trauma to the vascular smooth muscle stimulation trigger an immediate localized reflex contraction, as you can see over here. The smooth muscle fibers in tunica media constrict forcefully, narrowing the lumen and instantly reducing regional blood flow. Due to this contraction, blood flow is reduced right now. This rapid reflex action acts to buy critical time, significantly limiting initial blood loss and providing a crucial physiological window for more complex cellular and chemical cascades to deploy. Now let us introduce our second phase, the platylate plug formation. In this phase, we construct a temporary cellular barrier, which is called platylate plug. Under healthy conditions, the vessel lining is perfectly non-thrombogenic. That means under normal situation, blood vessel doesn't form any blood clot. But injury disrupts this boundary, exposing the structural subendothelial collagen fibers underneath. As you can see over here, due to injury, the subendothelial collagen is exposed. This raw collagen you see here serves as the physical foundation for repair. To bridge circulating platylates to this exposed collagen, a specialized plasma protein called von Willebrand factor or VWF binds tightly to the exposed strands. Now we will look at this diagram, which sees inside a blood vessel to further bridge the concept of hemostasis and von Willebrand factor. Under normal conditions, your blood flows smoothly through the blood vessel, and the blood vessel has a single layer of smooth cells called the endothelium, pointed with laser here. Hidden right beneath this protective lining is a layer of tough structural proteins called collagen. This is the collagen layer we are talking about. But when an injury happens, it creates an endothelial defect seen in this segment, which is a break in the wall. This tear exposes underlying collagen to the rushing bloodstream. This exposure is the universal distress signal that kicks off a two-part rescue mission. Phase 1 is called primary hemostasis, and it is all about building a temporary plug. A specialized protein called von Willebrand factor, indicated here, or VWF, which is bound to subendothelial tissue, immediately grabs passing cells called platylate. These are the platylates, and von Willebrand factor will capture them. To do this, platylates use a specific surface receptor called GP1B to anchor themselves to von Willebrand factor. This is GP1B, and with this anchoring site, it gets attached to von Willebrand factor. You can think of GP1B like a molecular velcro that allows platylates to stick firmly to the site of injury instead of washing away. Once anchored, these platylates become activated. They change shape and roll out a second set of receptors called GP2B or 3A. To build a strong barrier, platylates need to stick to each other, not just the vessel wall. They achieve this through a process called aggregation. A sticky plasma protein called fibrinogen acts as a molecular bridge, binding to GP2B/3A receptors on neighboring platylates and tying them all together into a tight network. As you can see, this platylate is getting attached to the platylate beneath, and by this, primary platylate plug is formed. But this is not the end. Finally, we will introduce you to the final phase, which is called the coagulation cascade. To understand coagulation cascade clearly, we will look at the previous diagram. This phase represents a highly complex series of protein activation steps where inactive plasma proteases are sequentially cleaved to activate the next factor in line, ultimately converging on a master regulatory target. The body triggers this cascade through two distinct primary pathways. The first one is the intrinsic pathway, initiated purely by contact activation inside the vessel. When circulating factor 12 hits the exposed subendothelial collagen, it changes shape and triggers a slower, highly amplified sequence. The second path is the extrinsic pathway, an explosive, rapid response triggered from outside the vessel wall. When tissue outside the vessel is physically damaged, it exposes a membrane glycoprotein called tissue factor, which binds directly with circulating factor 7 to kickstart a rapid activation sequence. Both intrinsic and extrinsic pathways converge directly on a singular point intersection, which is the common pathway. This convergence point initiates at factor 10, which forms a complex alongside factor 5 to create the prothrombinase complex. This active enzymatic engine executes a master biochemical conversion. It cleaves or breaks the inactive plasma protein prothrombin into the central enzyme of the entire cascade, thrombin. Thrombin then acts as a catalytic powerhouse, cleaving the large soluble plasma protein fibrinogen into insoluble thread-like strands of monomeric fibrin. These strands spontaneously become polymers, that means polymerize, wrapping around the loose platelet plug that was created earlier, like a reinforced mesh net that you see over here. To complete the architecture, thrombin activates factor 13, which physically cross-links the overlapping fibrin strands into a tight, stable, and permanent clot. Once healing is achieved, a localized enzyme called plasmin is introduced to dissolve the clot and restore smooth, uninhibited laminar flow. Now, to clear the confusion, let's recap the whole process under 1 minute in simple language. When a blood vessel is injured, the smooth muscle immediately contracts. This rapid vasoconstriction narrows the vessel lumen, drastically reducing blood flow to buy critical time for cellular repairs. Next, the damaged vessel exposes underlying collagen. This allows von Willebrand factor to bind and anchor circulating platelets to the injury site. Once attached, these platelets activate, releasing chemical signals like ADP and thromboxane A2 to recruit more platelets, forming a loose temporary plug seen over here. And to secure the plug, the coagulation cascade begins. It triggers through either the extrinsic pathway or the intrinsic pathway. Both pathways converge at the common pathway. Here factors 10 and factors 5 catalyze the conversion of prothrombin into the active enzyme thrombin. Thrombin then converts soluble fibrinogen into sticky fibrin bands or strands. These strands polymerize around the platelets like a net seen over here. Finally, factor 13 cross-links the strands, transforming the loose plug into a tight, stable, permanent clot. And when it is required, plasmin clears away the clot. Now that we have mapped out these mechanical reflexes, cellular bridges, and multi-pathway enzymatic cascades from first principles, let us transition directly to our text and anchor these concepts to the exact sequences and high-yield traps outlined on slide 13. First, hemostasis is defined as the strictly regulated physiological process responsible for the arrest of bleeding following a structural vessel injury. The hemostatic response operates as an integrated cascade consisting of 4 tightly linked milestones. First, vascular spasm, then platelet plug formation, the coagulation cascade, and finally fibrinolysis. To elaborate vascular spasm mechanism, this phase initiates as an immediate smooth muscle contraction within the injured vessel wall to drastically restrict local luminal diameter and reduce blood flow. Exposure of subendothelial collagen allows von Willebrand factor to form a molecular bridge which connects collagen with the GP1B receptor of platelet, triggering the activation of platelet, and as a result, platelet releases ADP and thromboxane A2 to build a loose plug via positive feedback. And this plug is called primary platelet plug. Then comes coagulation cascade. The chemical cascade splits into intrinsic pathway activated by factor 12 when it comes in contact with collagen, and an extrinsic pathway triggered by tissue factor and factor 7. Both regulatory pathways converge directly at factor 10 to activate the common pathway, which generates thrombin to convert soluble fibrinogen into insoluble fibrin strands. Then factor 13 cross-links the fibrin strands to make it stronger and solid. Once tissue repair is complete, the specialized enzyme called plasmin executes fibrinolysis, actively dissolving the fibrin clot to restore normal perfusion. Now we will pay attention to the high-yield exam traps and then exam pearls. A classic trap focuses on the precise target of corporate anticoagulant drugs. The drug name is heparin, and heparin functions by activating antithrombin 3, whereas warfarin explicitly blocks the synthesis of vitamin K dependent factors, which are 2, 7, 9, 10. For your information, there are in total 13 coagulation factors, and out of them, 2, 7, 9, and 10 are inhibited by warfarin. Mixing up these mechanisms or swapping their targeted factors is a standard way students lose points. Another important drug you need to know about is aspirin. Aspirin will inhibit cyclooxygenase enzyme, which will cause less production of thromboxane A2, and it will also decrease the platelet aggregation. That means aspirin can cause bleeding. Now, to seamlessly distinguish cascade pathways on complex exam stems, remember that intrinsic pathway represents a contact activation sequence occurring strictly inside the vessel, while the extrinsic pathway is driven by tissue factor originating from outside the vessel wall. Then finally, let me tell you about the thrombin amplification rule. To bridge chemical mechanics with clinical understanding, remember that thrombin functions as the ultimate multitasking enzyme. It not only produces the primary fibrin mesh, but also acts as a positive feedback multiplier or amplifier that accelerates upstream factors and activates factor 13 to secure the clot. The lymphatic system is an often underappreciated companion to the cardiovascular system, but its absence would be fatal within hours. So we will talk about lymphatic system in detail in this segment. To understand how the body cleans up extra fluid and moves fats, let us establish a conceptual map of the lymphatic system based entirely on the first principles. When the heart pumps blood into blood capillaries, the high pressure pushes water, small molecules, and some proteins out into the spaces between the cells. This spilled fluid is called interstitial fluid, or ISF. While most of this fluid goes back into blood capillaries on the low pressure venous side, a small amount is left behind every day. If this leftover fluid keeps building up, the tissues will swell dangerously. To fix this problem, we must introduce a specialized one-way cleanup network to gather this fluid and return it safely to the bloodstream. And that brings us to the lymphatic system. Let us position the start of this cleanup route on our conceptual map. As you can see, these are the tissues, and deep inside our tissues, right next to blood capillaries, these are the blood capillaries, we place lymph capillaries, color-coded green in this diagram. This is lymph capillary. These tiny tubes are completely blind-ended. You see that it has ended here. It doesn't have any connection after this point. So they are closed at one end like a dead end street. The walls are made up of overlapping cells that act like simple one-way swinging doors. When fluid builds up in the tissue, it pushes these doors open and flows inside. You see with this arrow, interstitial fluid is flowing inside the lymph vessels. And when it gets inside the lymph vessels or lymph capillaries, we will call it lymph. Now let us follow this fluid as it travels along larger lymph vessels. Remember, the fluid was collected over here and now it's flowing in this direction toward the large lymph vessels. And to keep the lymph moving in one direction toward the neck, we add tiny internal valves to prevent backflow. That means just like veins, lymph vessels will also have valves in it. Because the lymphatic system does not have a central heart to pump it, we must introduce an external squeezing force. We place skeletal muscles around the lymph vessels. That means skeletal muscles will be surrounding the lymph vessels, so that every time you move your body, the muscles squeeze the tubes and push the fluid forward. We also add a thin layer of smooth muscle in the walls of the larger vessels to help them contract. Now let's track the lymph vessel in the human body. Let's say this is the lymph vessel we're talking about, and lymph is flowing through this vessel. Before this fluid is allowed to return to the bloodstream, it must be thoroughly cleaned. For that, let us place specialized filter station along the path, which are called the lymph nodes. One lymph node is zoomed in this image. If you see clearly, then you will find that these lymph nodes are bean-shaped and small, which are packed with dense masses of white blood cells, especially lymphocyte and macrophages. These cells act as security guards, attacking any virus, bacteria, or abnormal particles traveling in the fluid. This diagram also includes other important defense organs such as the tonsil and adenoids in the throat, the thymus gland in the chest, where certain immune cells mature, and the spleen, which filters the blood itself. Now let us introduce an entirely separate and vital job for this drainage system, which is absorbing dietary fat. When you eat a fatty meal, your digestive system breaks the fats down into fatty acids and monoglycerides inside the lumen of intestine. As tracked on this image, these molecules are packed into tiny spheres called micelles, so they can enter the absorptive epithelial cells of the intestinal wall. You see that the micelles are entering inside the intestinal wall, and these are the intestinal cells having microvilli, which are finger-like projections. Inside these cells, the pieces, that is the micelles, are put back together into fats called triglycerides, and they are combined with proteins to form larger fat globules called chylomicrons. This is the chylomicron. Because chylomicrons are too large to fit through the tight walls of regular blood capillaries, we must place a specialized lymph capillary right inside the intestinal villi, which is called a lacteale. The chylomicrons can easily fit into the lacteale, allowing the lymph to carry the fat safely away from the intestine. Finally, all these lymph pathways converge into two massive main lines that dump the fluid back into the large veins of the upper body. The first main line is the right lymphatic duct, which collects fluid from the right upper side of the body and empties directly into the right subclavian vein. The second one, the much larger main line, is the thoracic duct. The thoracic duct collects all the lymph from the entire body below the diaphragm, plus the left upper side of the body, and carries it up to the left subclavian vein, safely returning the fluid and fats back to general blood circulation. Looking at the diagram, you might feel that the chylomicron went past the capillary, but actually it bypassed the capillary, then went inside the lacteole. Don't confuse that concept, because chylomicrons are too big to fit into the capillary. That's why they don't get inside the capillary. Rather, they accommodate into lacteals. Now that we have mapped out this one-way drainage route, the immune filter nodes, and that fat absorption pathways from principles, let us transition directly to our text and anchor these concepts to the exact definitions, numbers, and traps that we will find on slide 14. To formalize the slide, first we will talk about the lymphatic system, which is a one-way drainage network designed to return interstitial fluid and proteins back to the general circulation. This lymphatic system acts as a continuous safety valve, draining approximately 3 liters of leaked capillary fluid every single day. The initial collecting tubes are called lymph capillaries, which are completely blind-ended and are positioned directly within the body tissues to gather extra fluid. Lymph nodes serve as local filtration stations that clean the lymph and act as major sites for active immune responses against pathogens. Then comes thoracic duct. This is the major drainage highway which collects lymph from the entire body below the diaphragm and left upper half of the body, dumping it straight into the left subclavian vein. Then the right lymphatic duct, which is the smaller main tube, it drains fluid from the right upper half of the body, emptying it directly into the right subclavian vein. Lacteals are specialized lymph capillaries located within the intestinal villi that function specifically to absorb dietary fat in the form of chylomicrons. Be aware of the exam traps that I am about to mention. The first trap focuses on the condition known as lymphedema. If a lymph vessel is blocked, proteins begin to accumulate in the tissue spaces. According to the rule of osmosis, this accumulation raises the interstitial osmotic pressure, which actively draws more water out of the blood and into the tissues, causing severe localized swelling. And classically, you will see the swelling in the legs of the patients. This is a patient of lymphedema where you see the right leg is normal, but the left leg is swelled. Another major multiple choice trap targets the exact pathway of fat transport from a meal. Question options will try to trick you into stating that digested fats enter the hepatic portal vein to go straight to the liver like proteins or sugars do. Remember the strict rule highlighted on this slide. Fat absorption does not enter portal vein. Instead, it enters local lacteals, moves through the lymph, travels up the thoracic duct, and empties directly into the blood via the subclavian vein. On high-yield anatomy questions, remember that lymph vessels are structurally very similar to veins because they contain internal valves to stop backflow, and they rely on the squeezing of surrounding skeletal muscle and smooth muscle contraction to push fluid forward. Finally, to bridge this anatomical layout with your exam questions, always remember that the thoracic duct drains a much larger portion of the body than the right lymphatic duct, making the left subclavian vein the primary re-entry point for clean lymph and absorbed dietary fats. This is the thoracic duct we were talking about. Its journey begins in the abdomen, about here, and from this place, the single vessel climbs straight up your chest cavity, squeezing right behind the esophagus and running parallel to your spine. Along the way, networks of intercostal glands act as filters that you see here. Finally, at the base of the neck, over here, the duct empties its payload directly back into your bloodstream at the junction of the left internal jugular and subclavian vein. That means here. This is the subclavian vein and this is the jugular vein. And without this crucial loop recycling fluid every single day, your cardiovascular system would fail. To conclude our lecture slides before moving to the summary table, let us establish a foundational map of blood components to see how the fluid system is organized. Remember, whole blood is not a uniform liquid. It is a specialized connective tissue consisting of living cellular entities suspended within a complex non-living fluid matrix. Let's say this is a test tube, and inside this test tube you have blood. Now if you spin this test tube using a centrifuge, the mechanical force separates the mixture into 3 distinct layers, purely based on the physical densities of its components. Let us position the uppermost least dense layer on our conceptual map, and that is plasma. Making up the largest portion of the whole blood, plasma functions as the liquid highway of the body. It is composed almost entirely of water, which acts as a universal solvent to dissolve and transport vital solutes, including nutrients, metabolic waste, hormones, and dissolved gases. Suspended within this water matrix are specialized plasma proteins manufactured primarily by the livers. Now let us introduce 3 essential classes of these proteins to understand their hemostatic utility. First, we place albumin, the most abundant plasma protein. Albumin is too large to easily pass through capillaries, so it stays in the blood vessel to keep water from leaking out into tissues. Second, we position globulins, which serve a dual purpose. Some function as alpha and beta transport proteins, while others form gamma globulins, also known as immunoglobulins, which are antibodies built for target immunity. Third, we add fibrinogen, a vital soluble clotting factor that stands ready to turn into solid strands during an injury. Now let us move down the test tube to introduce the heavier solid elements that settle out below the plasma. Collectively, these are defined as the formed elements. Immediately below the yellow plasma, we find a very thin cream-colored intermediate layer known as the buffy coat, which is clearly illustrated in this image. We place 2 specific types of formed elements here. The nucleated white blood cells, or leukocytes, which provide our primary immune defense, and the cell fragments or platelets, which are built to handle immediate hemostasis. Finally, at the absolute bottom of the tube, we place the densest and most numerous formed elements, which are the red blood cells or erythrocytes. Because these cells are packed with iron-dense hemoglobin, they sink to the bottom during spinning to form a deep red layer. This specific fraction of red blood cells is called hematocrit. The primary job of these cells is to pick up oxygen in the lungs and distribute it to body tissues. Let us now look at how the proportion of this red layer changes under different physiological conditions. In a healthy individual, the hematocrit level stays within a strict homeostatic range. However, if the body fails to produce enough red blood cells or loses them rapidly, the red layer shrinks significantly, that you see in the second test tube. And this state of depressed hematocrit percentage is called anemia. Conversely, if the body produces too many red blood cells, the red layer expands, that you see in the third test tube, that it is expanding. This state of elevated hematocrit percentage is called polycythemia, which makes the blood thick and harder to pump. Now that we have mapped out this layered composition, we can transition directly to our text and anchor these concepts to exact definitions, normal ranges, and clinical traps. As usual, we start with the defining points. Whole blood is divided into two main fractions: the liquid plasma, which makes up about 55% of blood, and the solid formed elements, which account for the remaining 45%. Plasma is a complex fluid made up of 90% water, along with plasma proteins, dissolved nutrients, hormones, gases, and electrolytes. The main three plasma proteins are: first comes albumin, and this protein works specifically to maintain the colloid osmotic pressure of the plasma, keeping fluid inside vessels. Then comes globulins. These proteins are responsible for crucial immune functions as immunoglobulins and act as transport molecules for lipid and ions. Then comes fibrinogen. This protein acts as a key clotting factor that is completely absent in serum samples. Then we will talk about the formed element subtypes. First comes erythrocyte. These red blood cells make up bulk of the hematocrit layer and are dedicated strictly to systemic oxygen transport. Leukocytes are the white blood cells that settle within the buffy coat layer to handle active immune defense. That means white blood cells are also known as leukocytes. Then comes platelets. The cell fragments rest alongside the white blood cells in the buffy coat to manage hemostasis after a blood vessel injury. Never get confused with the exam traps I'm about to mention. The first confusion that the examiners will try to create is by asking you to find a sample containing all the raw proteins of a blood clot. Remember the explicit trap highlighted on this slide. Plasma contains the intact protein fibrinogen, while serum does not contain clotting factors because they are completely removed when the sample is allowed to clot. Another major trap focuses on the clinical consequence of starvation or liver disease. If the liver cannot produce enough albumin, the body experiences a low albumin level. This drop reduces the plasma osmotic pressure, which means the blood can no longer hold onto its water, causing fluid to leak out into the surrounding spaces to produce tissue swelling or edema. On high-yield biology questions, remember that the term hematocrit is strictly defined as the percentage of the total blood volume occupied by the red blood cells. And to bridge this measurement with clinical states on your exam, remember that the hematocrit value is elevated in patients with polycythemia and significantly reduced in patients suffering from anemia. We have covered enormous ground in this lecture. Let me give you a rapid walkthrough of the highest yield facts, the ones most likely to appear on your IMAT paper. I also want to give a massive thank you to every single one of you who stayed this long and stuck with us through this entire deep dive. Your dedication to mastering these first principles is exactly what will set you apart on exam day. Let us use this final master anchor slide to lock in all the core parameters, anatomical layers, and physiological rules we covered so you can protect your score. Reviewing our master structure information. The heart functions as four-chambered dual muscular pump. Its walls are split into the protective outer epicardium, the heavy contractile myocardium, and the ultra-smooth internal endocardium that prevents dangerous blood clots. To keep blood moving strictly forward, the heart relies on four passive valves. The tricuspid valve handles the right side, having three cusps. The mitral valve or bicuspid valve handles the left side, having two cusps, and the pulmonary and aortic semilunar valves guard the main arterial exits. When cardiovascular efficiency is measured by specific volume markers, the maximum fluid the ventricle holds after filling is the end-diastolic volume, or EDV, which averages around 130 milliliter. The residual fluid left behind after pumping is the end systolic volume, or ESV, averaging 50 to 60 ml. The absolute volume pushed out per beat is the stroke volume, calculated using the explicit formula. That is, stroke volume is equal to end diastolic volume minus end systolic volume. And multiplying this stroke volume with heart rate gives the total volume moved per minute, which is the cardiac output. And pumping efficiency is tracked as a percentage using the ejection fraction formula. EF, or ejection fraction, is equal to stroke volume over end diastolic volume times 100, which will give the result in a percentage. And in the isovolumetric phase of the cardiac cycle, all four valves will be closed. Now the blood vessels. Your circulatory system relies on three main types of vessels, each adapted for a specific job. Arterioles serve as the primary site of vascular resistance, meaning they constrict or dilate to control blood flow into tissues. Capillaries are ultra-thin and act as the primary site for nutrient and gas exchange. Veins work quite differently. They have high compliance and serve as a reservoir, holding about 60 to 70% of your body's total blood volume at any given time. If you look at their structure, standard vessels have three distinct layers: the tunica intima, tunica media, and tunica adventitia. Capillaries are the exception here. To maximize exchange efficiency, they consist of the tunica intima only. Next up is the blood itself. Red blood cells, or erythrocytes, are highly specialized. They mature without a nucleus or mitochondria, which means they rely entirely on anaerobic glycolysis for energy. They circulate for a lifespan of about 120 days. Inside these cells, you find hemoglobin, a protein made of four subunits, four heme groups, four ions in the ferrous state. This structure allows a single hemoglobin molecule to carry a maximum of four oxygen molecules. While oxygen binds directly to hemoglobin, carbon dioxide travels differently. About 70% of it is converted and transported in the blood as bicarbonate ions. Finally, remember a quick biochemical distinction. Serum is simply blood plasma with the clotting factors removed. Now let's look at how the body regulates blood pressure. The fundamental equation to memorize is that blood pressure equals cardiac output multiplied by peripheral vascular resistance. The body uses two main systems to keep these in balance. First, baroreceptors provide fast neural response to immediate pressure changes. Second, the Renin-Angiotensin-Aldosterone system, or RAAS, provides slow hormonal response. In the RAAS pathway, the kidneys release renin, which leads to the formation of angiotensin 1, and as it passes through the lungs, angiotensin converting enzyme converts it into angiotensin 2. Angiotensin 2 then triggers powerful vasoconstriction to narrow the vessels and stimulates aldosterone release to retain salt and water, both of which raise blood pressure. To wrap things up, let's highlight three major exam traps where students frequently lose points. First, do not assume arteries always carry oxygenated blood. The pulmonary artery carries deoxygenated blood from the heart to the lungs, while the pulmonary vein carries freshly oxygenated blood back to the heart. Second, because red blood cells lack mitochondria, they cannot perform aerobic respiration. This means they do not consume any of the oxygen they are transporting. Third, remember how nutrients enter the portal vein. dietary fats bypass it entirely. Instead, fats enter the lymphatic vessels, which are called lacteals, and travel up through the thoracic duct and empty directly into the subclavian vein. Keep this core principle and exceptions in mind, and you will be well prepared for any question on cardiovascular physiology or the circulatory system. Let's transition straight into our high-yield question solving session. We have a total of 10 practice questions to master. So let's jump right into question 1, which focuses on the mechanics of the cardiac cycle. The question asks, during isovolumetric contraction of the left ventricle, which of the following correctly describes the state of the heart valves? To tackle this efficiently, let's break down the word isovolumetric. Iso means same and volumetric refers to volume. This tells us right away that during this specific phase, the volume of blood inside the left ventricle remains completely constant. For blood volume to stay exactly the same while the muscular walls of the ventricle are actively contracting and building up immense pressure, there cannot be any blood entering or leaving the chamber. So let's say this is the heart and we have 4 chambers. These are atria and ventricle. So the blood inside the ventricle, they cannot leave the ventricle. Otherwise, it will not remain isovolumetric. Now let's trace the movement. Before this phase, the left ventricle was filling with blood from the left atrium through an open mitral valve. As the ventricle fills completely and begins to contract, the pressure inside the ventricle instantly shoots up, rising higher than the pressure in the atrium. This sudden pressure reversal forces the mitral valve to slam shut to prevent any blood from leaking backward. This closure creates the first heart sound or lub. Now the ventricle is contracting against a sealed entry. But what about the exit? The blood wants to push forward into the aorta through the aortic valve here. However, at the very start of contraction, the ventricular pressure hasn't yet surpassed the high resting pressure inside this aorta. Because the pressure inside the aorta is still higher, the aortic valve remains firmly shut. This is the aortic valve we were talking about. Since the mitral valve has just closed and the aortic valve has not opened yet, the left ventricle becomes a completely sealed chamber. The heart muscle squeezes, pressure skyrockets, but the blood volume cannot change. Therefore, both the mitral valve and the aortic valve must be closed. Looking at our options, this perfectly matches option D. Moving right along to question 2. This question targets one of the major exam traps we emphasized earlier regarding the direction and oxygenation status of blood in pulmonary circulation. The question asks us to identify the errors in a student's statement. And the statement is, pulmonary arteries carry oxygenated blood from the lungs to the heart. Let's dissect this statement by applying 2 fundamental rules of cardiovascular anatomy. Rule number 1 concerns the direction of blood flow. By definition, all arteries carry blood away from the heart, while veins return blood back to the heart. Therefore, any vessel labeled an artery must be moving away from the heart ventricles, not toward it. And rule number 2 concerns the pulmonary circuit specifically. Blood leaving the right side of the heart is depleted of oxygen because it is returning from the body tissues. The heart pumps this deoxygenated blood. That means right ventricle will pump this blood through the pulmonary arteries out to the lungs so it can drop off carbon dioxide and pick up fresh oxygen. When we look back at the student's statement, they actually made 2 distinct errors. First, they claim that pulmonary arteries carry oxygenated blood. In reality, because this blood is on its way to the lungs to pick up oxygen, it is completely deoxygenated. Second, they stated that these vessels travel from the lungs to the heart because they are arteries. They must travel away from the heart toward the lungs. These vessels that actually carry oxygen-rich blood from the lung back to the heart are actually pulmonary veins. By combining this correction, we can see that pulmonary arteries carry deoxygenated blood and they carry it away from the heart toward the lungs. Scanning through our choices, This exact pair of corrections is beautifully summarized in option B. Let's advance to question 3, which explores the electrical conduction system of the heart. The question asks which component of the cardiac conducting system introduces a delay in impulse transmission, allowing ventricular filling to complete before ventricular contraction begins. The correct answer is option B, the AV node or atrioventricular node. If we draw this heart, AV node is located in the lower interatrial septum, and the AV node acts as an electrical gateway between the atria and ventricles. Its cells have a smaller diameter and fewer gap junctions, which naturally slows down the electrical signal by about 0.1 seconds. This brief pause is absolutely critical for proper mechanics. It ensures the atria have enough time to finish contracting and completely top off the ventricles with blood before those larger ventricles begin squeezing. To solidify this concept, let's quickly break down why the other options are not correct. Option A, SA node, is wrong because sinoatrial node is the heart's natural pacemaker. Its job is to initiate the action potential that sets the heart rate, not delay it. Option C, bundle of peas, is incorrect because it functions as an electrical bridge through the fibrous skeleton of the heart, rapidly conducting the signal downward from AV node into septum. Option D, right bundle branch, is wrong because it is simply a pathway that splits from the bundle of His to rapidly distribute the impulse deep into the right ventricular wall. Purkinje fiber, or option E, is incorrect because these specialized fibers are built for speed, not delay. They distribute the signal to the ventricular muscle cells incredibly fast to trigger a coordinated, powerful contraction from the apex upward. Therefore, AV node is uniquely specialized to introduce that vital mechanical delay, making option B our definitive answer. Moving along smoothly to question 4, which asks us to identify a property that is unique to mature human erythrocytes when compared to other blood cells. And the correct answer is option C. They lack a nucleus and mitochondria. During their development in the bone marrow, human red blood cells undergo a highly specialized process where they extrude their nucleus and break down their organelles, including mitochondria. This complete lack of nucleus and mitochondria is a defining anatomical feature unique to mature erythrocytes among the actual true cells found in our blood, leaving the cell's interior almost entirely packed with hemoglobin to maximize its oxygen carrying space. Let's carefully evaluate why the other options do not satisfy the criteria for a property unique to erythrocytes compared with other blood cells. Option A, they contain hemoglobin, and option E, they transport oxygen, are closely related functional properties. While hemoglobin and major oxygen transport are indeed the primary characteristics of erythrocytes, these options describe what they contain and do rather than a structural cell biology trait that distinguishes their cellular architecture uniquely from all other whole blood cell populations. So they are not correct. Option B states they are produced in the bone marrow. This is incorrect because this trait is shared by virtually all blood cells. White blood cells and the precursors or platelets are also produced via hematopoiesis within the rat bone marrow. Option D, the lifespan of them are approximately 120 days. This is wrong because while it is a specific metric for red blood cells, a specific lifespan duration itself is not a unique structural property of a cell line, and other cell types have their own specific operational timelines. Thus, the structural adaptation of operating as an anucleate cell devoid of mitochondria makes option C the standout characteristic unique to mature erythrocytes. We are halfway through our practice session with question 5, which bridges basic anatomy with real-world clinical pathology. The question presents a scenario involving a patient who has the stenosis of mitral valve and asks, when the pressure difference between the left atrium and left ventricle is greatest, which heart sound or acoustic phenomenon would be effective. To understand why, let's look at when the mitral valve is supposed to be doing its job. Under normal conditions, the mitral valve, which is situated between the left atrium and left ventricle, opens wide during ventricular diastole, which is the relaxation and filling phase of the cardiac cycle. This allows blood to flow smoothly and effortlessly from the left atrium down into the left ventricle. However, when a patient has mitral stenosis, that valve opening becomes significantly narrowed and restricted. That means the valve will not open as wide as before. Because the opening is small now, it acts like a bottleneck, creating massive resistance to blood flow. To force blood across this tight barrier, the left atrium must squeeze much harder, causing the pressure in the left atrium to skyrocket, while the pressure in the relaxing left ventricle remains comparatively low. This creates a large abnormal pressure gradient between the two chambers precisely during ventricular filling. Furthermore, forcing blood through this narrowed, rigid opening turns normal, smooth laminar blood flow into highly turbulent flow. In medicine, a turbulent blood flow across a damaged or narrowed valve is heard through a stethoscope as a murmur. Since this turbulence happens while the ventricles are filling during diastole, it creates a characteristic diastolic murmur. So the correct answer would be option C. During ventricular filling or diastole, a diastolic murmur would be present. Let's briefly rule out the other options to ensure a complete understanding. Option A and B are incorrect because during ventricular contraction and ejection, the mitral valve is supposed to be completely closed anyway. The pressure differences during these phases are governed by ventricular contraction and aortic dynamics, not by a stenosis resisting forward filling flow. Option D is incorrect because isovolumetric relaxation is a brief phase where all valves are closed before filling begins. So filling murmur wouldn't be defined here, nor would it cause S2 to be absent. Option E is wrong because while atrial systole is part of late diastole, the pressure difference and turbulent flow span across the entire ventricular filling phase, and it doesn't cause the S1 sound to completely disappear, but it says that it is absent. That's why it would be wrong too. Therefore, option C provides the most accurate physiological and clinical description of mitral stenosis. Let's break down question 6, which tests your understanding of the hormonal regulation of blood pressure via the endocrine cascade known as RAAS. The question reads, the Renin-Angiotensin-Aldosterone system is activated when blood pressure falls. Which of the following sequences correctly describes the cascade? To clearly get the answer and explanation, first talk about what happens when our blood pressure or blood volume drops. When this happens, let's say blood pressure or volume is dropping, the juxtaglomerular cells in the kidneys sense this change and immediately release an enzyme called renin into the bloodstream. Renin's specific job is to locate a large plasma protein constantly produced by the liver, which is called angiotensinogen, and cleave it down into an inactive 10 amino acid peptide known as angiotensin 1. As angiotensin 1 circulates through the small capillaries of the body, particularly within the lungs, it encounters an endothelial enzyme called ACE, or angiotensin converting enzyme. ACE rapidly cleaves off 2 more amino acids from angiotensin 1, converting it into the highly active hormone angiotensin 2. Angiotensin 2 is a potent vasoconstrictor that binds to blood vessels to narrow them and increase vascular resistance. At the same time, angiotensin 2 travels through the blood to the outer layer of the adrenal glands, specifically the adrenal cortex, where it binds to receptors and triggers the synthesis and release of aldosterone. Aldosterone then acts on the kidneys to conserve sodium and water, which expands blood volume and restores blood pressure back to normal homeostatic level. Now let's cross-reference this exact physiological sequence against our choices. Option A is incorrect because renin converts angiotensinogen to angiotensin 1, not directly to angiotensin 2. Option B is incorrect because ACE converts angiotensin 1 to angiotensin 2, not renin. Option D gets the order completely backward, claiming ACE acts first on angiotensinogen. Option E inaccurately states that aldosterone stimulates renin release, which is the reverse of how the feedback loop operates. Tracing the sequence perfectly from renin cleavage to ACE conversion and finally to aldosterone secretion, we arrive at the correct answer, which is option C. Now let us carefully analyze question 7, which shifts our focus to the microanatomy of different blood vessels and how their structural dimensions directly relate to their physiologic function. The question asks, which of the following vessels has the thickest tunica media relative to its total wall thickness and why? To solve this, let's look at the phrase relative to its total wall thickness and why. While large elastic arteries like the aorta contain the absolute greatest amount of elastic tissue and overall wall thickness to withstand and buffer high systolic pressure, we have to look closely at the proportional layout of the smaller vascular branches. Let's say this is a big artery. As this artery branch out and gets smaller, their mechanical requirements change from merely stretching to actively regulating downstream blood distribution. When we look at arterioles, their total vessel wall is incredibly thin compared to a massive artery, but that wall is composed almost entirely of concentric layers of smooth muscle making up the tunica media. Proportional to their tiny diameter and total wall thickness, arterioles have the highest percentage of smooth muscle tissue. This specific structure composition is critical because arterioles function as the body's primary resistance vessels. Their heavily muscular tunica media, if you draw and zoom in, let's say this is an arteriole, you will see that the tunica media is massive and filled with smooth muscle. Let's say this is the layer. Their heavily muscular tunica media allows them to alter their diameter dynamically via vasoconstriction and vasodilation in response to neural, hormonal, and local chemical commands, which dictates minute-to-minute blood flow into capillary beds and stabilizes systemic blood pressure. Let's double-check why other choices don't fit, apart from option D. Option A and option E are incorrect because veins and venules operate under extremely low pressure and possess a very thin, poorly developed tunica media relative to their total wall. Option B is fundamentally incorrect because capillaries do not have a tunica media at all. They consist solely of a single layer of endothelial cells to allow for rapid fusion. Option C accurately describes the function of large elastic arteries such as aorta, but their wall composition contains an abundance of elastic sheets interspersed throughout, meaning the smooth muscle component within the tunica media is not as dominant relative to total wall thickness as it is seen in an arteriole. Thus, looking for the vessel whose wall is proportionally dominated by smooth muscle for vascular resistance points us to the final answer being option D. Let's dive into question 8, which explores the mechanical relationship between a respiratory system and cardiovascular hemodynamics. The question states, during inspiration, thoracic pressure decreases. Which effect does this have on venous return and cardiac output? To figure this out, let's look at the physical mechanics of breathing in. When you inspire or when you take a breath in, your diaphragm will contract and it flattens downward. That means, let's say if this is the normal position of the diaphragm, it will flatten out like this. At the same time, your external intercostal muscles lift the ribs upwards and outward. This expansion significantly increases the volume inside your chest or thoracic cavity. According to Boyle's law, as volume increases, pressure must drop, meaning the intrathoracic pressure becomes negative or subatmospheric. At the exact same time, because the diaphragm is pushing downward, as we have seen in this illustration, this diaphragm compresses the organs and blood vessels within your abdominal cavity, causing intra-abdominal pressure to rise. Now, look at the pressure gradient created between these two body cavities. Fluid naturally flow from areas of high pressure to areas of low pressure. Because the abdomen has high pressure and the thorax has now low pressure, this difference acts just like a biological vacuum or syringe plunger. It sucks blood out of the large lower body veins, such as the inferior vena cava, and draws it up into the thoracic cavity and into the right atrium of the heart. This vital mechanical process is known as the respiratory pump. Because more blood is actively drawn back to the heart during inspiration, the venous return will increase. And due to Frank-Starling law of the heart, this increased filling volume stretches the heart muscle, resulting in a more powerful contraction, which ultimately will boost stroke volume and cardiac output. And this description matches perfectly with option B, which will be our correct answer. Now let's review why other options are incorrect. Option A is wrong because the pressure gradient is not reversed. It is actually enhanced in a way that favors upward flow. Option C is incorrect because while venous valves prevent blood from slipping backward due to gravity, they do not block the forward suction force of the respiratory pump. Option D is incorrect because cardiac output increases and the lower thoracic pressure actually reduces the resistance the right ventricle faces when pumping blood into the lungs. Option E is false because the respiratory system exerts a massive immediate impact on venous dynamics. By matching our understanding of the respiratory pump pulling blood upward, we can confidently identify the correct choice, which is option B. Let's break down question 9, which focuses on a classic physiological trap regarding how the body absorbs fat. The question reads, which of the following is the correct site of fat absorption and transport in the digestive circulatory system? To understand this question and get to the answer, first let's check our knowledge. When you consume carbohydrates or protein, they are broken down into simple sugars, that is glucose and amino acids. They are small enough to pass directly into the blood capillaries within the intestinal villi. From there, they travel straight to the liver via the hepatic portal vein. Dietary lipids, however, undergo a completely different pathway due to their chemical nature and size. Once inside the intestinal epithelial cells, that means the enterocytes, this is an enterocyte, the fatty acids and monoglycerides are reassembled into triglycerides. These triglycerides are then packaged together with cholesterol and special proteins to form large spherical lipoprotein particles called chylomicrons. Because chylomicrons are incredibly large, they cannot physically squeeze through the tight junctions or the continuous basement membrane of the standard blood capillaries found in the intestinal villi. Instead, they must enter a specialized, highly permeable lymphatic capillary located in the center of each villus. If we zoom into one of these villus, you will see it will have a lymphatic capillary located in the center, which we will call lacteal. Once this chylomicron enters inside the lacteal, they travel through lymphatic vessels, passing through the main lymphatic trunk known as the thoracic duct. The thoracic duct then empties this fluid directly into the venous bloodstream at the junction of the left subclavian and internal jugular veins. This unique anatomical routing means that dietary fats completely bypass the liver and the hepatic portal system during their initial absorption. By tracing this unique route from intestinal villi to lymphatic vessels and finally into the systemic circulation, we find the description that aligns perfectly with reality in option B. Now let's evaluate the incorrect choices based on this pathway. Option A and option D are incorrect because they claim fat are absorbed directly into blood capillaries or hepatic portal vein, which only happens for small water-soluble molecules. Option C is incorrect because free fatty acids binding to albumin is a mechanism used when the body mobilizes stored fat out of adipose tissue during fasting, not during dietary absorption from the gut. Option E is false because chylomicrons are assembled inside the intestinal epithelial cells, not downstream inside the lymph nodes. We have made it to our 10th and final question, which tests your ability to apply core cardiovascular formulas to solve a quantitative clinical scenario. The question reads, a 70 kilogram man has resting heart rate of 72 beats per minute and an end diastolic volume of 130 milliliter. If his ejection fraction is 60%, what is his cardiac output? To solve this step by step, we need to connect three fundamental parameters of cardiac performance: heart rate, stroke volume, and ejection fraction. Our ultimate goal is to find cardiac output, which represents the total volume of blood pumped by heart per minute. The formula for cardiac output is CO is equal to heart rate times stroke volume. We are already given the heart rate, which is 72 beats per minute. However, the exact stroke volume is not directly provided. Instead, we are given the end diastolic volume and the ejection fraction. Let's calculate the stroke volume first from that information. The end diastolic volume we see is given 130 ml. The ejection fraction is 60%. Now to find the actual blood volume ejected, that means stroke volume, we multiply the total volume by the percentage pumped out. That means stroke volume would be equal to end diastolic volume times ejection fraction. 60% or 0.60, that is 78 milliliter. Now that we know the heart pumps out exactly 78 milliliter of blood with every single beat, we can plug this value back into our primary equation to calculate the total output per minute. So stroke volume given 78, if we multiply them both, will be around 5616 milliliter per minute, which exactly matches with option B, giving us the correct answer. That wraps up our complete deep dive into the human circulatory system. A massive thank you to every single one of you who stayed with us until the very end of this lecture. Your dedication to mastering these concepts from absolute first principles is exactly what is going to give you an edge on exam day. If you want to take your preparation to the next level and keep all of these high-yield concepts right at your fingertips, make sure to check out our biology living notes. These notes are fully updated, beautifully structured, and specifically designed to map out exactly what you need to know for the IMAT without any fluff. You can find the link to grab your copy right down in the description below. And if you found this session helpful, please make sure to like this video, subscribe to this channel, and hit that notification bell so you never miss a high-yield medical entrance topic. I also want to address why it takes such a long time to get these videos out to you. Building these comprehensive high-yield lectures completely from scratch and ensuring every single mechanism is explained with absolute clinical perfection takes an immense amount of research, planning, and detailed production time. We refuse to cut corners on your education. However, we know you want more content faster, which is why we are officially hiring video editors right now. And we have recruited some talented guys over here. Finally, thank you again for your incredible support. Keep studying smart, and I will see you all in the next video for our next high yield session.